Speaker profile. Speaker profile.
I request all the delegates to assemble in the auditorium.
Engineer
Jai Shri Gurudev.
By seeking the blessings of His Holiness Paramapujya Jagat Guru Sri 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 Dr. Balagangadhara Tama Swamiji and rendering our humble pronouns to Paramapujya Sri 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 Dr. Nirmalanandana Tama Swamiji, we welcome you all to the second day of the International Conference on Innovation and Advances in Pharmaceutical Sciences organized by Sri Adichunchanagiri College of Pharmacy together with Association of Pharmaceutical Research APR.
This is Lohita.
And this is Rakshit.
It gives us immense pleasure to host today's event as we bring to you this morning filled with desires, hopes, and dreams we all share.
For the first keynote session of the day, we have Dr. Rajesh Venkataraman, Professor and Head, Department of Pharmacy Practice, Sri Adichunchanagiri College of Pharmacy, Adichunchanagiri University.
Sir is here to speak on advancing healthcare, the power of artificial intelligence in the pharmaceutical industry.
Ladies and gentlemen, put your hands together for Dr. Rajesh Venkataraman.
Jai Shri Gurudev.
I'm Dr. Rajesh Venkatraman, currently serving as Head Department of Pharmaceutical Sciences. At the outset, I would like to start by showing my gratitude to the organizers. A special thanks to you, sir. Special thanks to Dr. Prakash Gaudanawar, and special thanks to Dr. Ramesh to have considered me, enough to share the podium with eminent speakers and who are, hallmarks in the profession. Thank you so much for your consideration, sir. Thank you. I'm here to deliberate and visualize the future of pharmacy around the globe, enabling you to embrace the advancing healthcare through digital transformation. Okay. To digital transformation for better patient care. I suppose things started with that of the onset of the pandemic that has brought changes and stretched the personal and professional resilience to the limits. The results have embarked us never before working into closely as professionals working and embracing the technology. So it was not long since the start wherein the Google giant, that is when they saw that there were 7% of Google searches that were being health related, immediately the health, when the search engine giant health, Google, they started to work with the healthcare through their healthcare data engine, tele and virtual health, remote work, healthcare analytics and insights, patient services and support, healthcare API management, patient app management, frontline caregiver devices, healthcare operational dashboards, visualization of healthcare services, smart factory AI, data analytics. So with this, it's not just Google that has set the frame, it was also Amazon. They started working from the year 2021 towards the forecast, artificial intelligence in medicine, digital health research, future of medicine, future of pharma, health insurance, healthcare design, and so on. So for your information, Amazon dives into healthcare through Amazon's project called as Cooper. It's an upcoming low Earth orbit that is called as LEO, satellite internet services that will be provided in the whole country through launching of 1,500 satellites. So basically to reach the rural parts of the globe. Fine. So I don't think there will be any issues with the rural part taking up for the healthcare research. So as you people know, Apple had worked on these things, and we just were thinking it has come over, just a year, but they were working for more than a decade or so for bringing in all these things. Apple has partnered with the global star for a satellite operation, and it plans on SpaceX. It's basically a satellite of their own to create its own emergency communication services. Apple's emergency SOS via satellite, which will be initially navigation kind. So this has killed navigation giants such as Garmin. People who are using iPhone 14, you may be having an option called as SOS that works directly with satellite. So it has been provided free of charge, wherein Garmin charges for providing these services. So that's how Apple has started working. So the moves of Google, the moves of Amazon, the moves of Apple, they all provide insights for us for the trends adopted by those companies which are in fact early predictors for the healthcare providers to embrace technology transformation in healthcare. So that's what I mean to say when these giants have embraced change, it's a high time we as healthcare providers start embracing these changes. So with this small introduction, I would like to start my session. Today we'll be brushing up more towards artificial intelligence. We had a lot of speakers speaking about artificial intelligence, though I'll be speaking related to artificial intelligence and healthcare, and that is most patient specific. We'll be speaking about 3D printing in healthcare, augmented intelligence, robotic process automation, big data analytics in hospital pharmacy settings. So artificial intelligence in pharma refers to use of automated algorithms to perform traditional small human intelligence oriented services tasks. So over the past like 5 years or so, artificial intelligence in pharmacy industry has redefined itself, and even in the drug development and clinical trial area, it has worked wonders. The probability of success of clinical trial, it relies basically on the biopharma investors, and they need to evaluate when making scientific and economic decisions on how you recruit patients. And artificial intelligence works enormously, wherein you get data in your fingertips, wherein it boosts the morale of the investors, because they do have the data on when they can complete the trial. It also allows biggest challenges, that is estimating the success rate of clinical trial, and also is so accurate that humans cannot provide. Gathering such data is very time consuming, and as you guys know, it's more susceptible to a lot of human errors. So the use of natural language processing, they simplify and they extract data, and they give this data to the pharma industries, wherein they come up with some decisions, and decision is making is what is making the decrease in timelines for the drug manufacturing. A staggering 86% of clinical trials fail because of the recruitment problem. Eventually, when you use artificial intelligence, this 86% can drastically decrease into 20% now, because you have the data of where the patients fall in inclusion criteria, exclusion criteria, and all those things. Fine. So artificial intelligence is a sophisticated pattern of recognition. Okay, it recognizes things. For example, I would like to quote, it's just a study in cancer patients provides across to personal and therapies to their genetic makeup and lifestyle. Yesterday we had a speaker who was speaking about genetics. So artificial intelligence gives the specific genetic codon, wherein the artificial intelligence through its computer designed program can help the clinical pharmacist, the doctors, come up with accurate, best drugs that are economic and that do not cause a lot of side effects. So artificial intelligence, yes, it works a lot in the healthcare sector, and especially in terms of prescribing and patient safety. What more is keen to the pharmaceutical and biotechnology companies are using machine learning algorithms to shorten the drug development process. As yesterday you would have seen many speakers speak that this artificial intelligence basically brings down the drug development process timelines, and this is more important wherein the public is being given new drugs, and with that of COVID, the requirements for a new drug that to hit market as early as possible. That's what it is. Right? So you must be knowing ChatGPT that has hit the globe recently, but we had such kind of things way back in 2021, where this is research being done, and this is a software kind that was used in China. So you can see basically we do the patients were using this artificial intelligence embedded chatbot system, wherein they can directly question the software, and the software will keep answering them according to what they require, enabling them diagnose. Even they diagnose, so you can also give your laboratory investigations for concise, concise diagnosis. So if you're not happy with the concise diagnosis, it will pave way for better treatment through some more referrals also. So you can see Dr. Board interface. You can see it's all been translated from that of Chinese. And you can see the end part. So they've concluded by giving a diagnosis, and they are also providing direct reports. So it's how fast things are. And if you see here, this is the same study. So basically, these are more useful in case of diseases which are stigmatized. Some patients who are having dermatological issues, sexually transmitted disease issues, things that are stigmatized, they are not comfortable to speak. So these chatbots work well with such kind of diseases that don't require major intervention, such as emergency treatment, and so on. So that's how things are. So, but it's not way back that we start believing each and everything. So there are cautions with AI-generated contents in biomedicine. So there are a lot of researchers who are cautioning that the data may not be precise. So it's a matter of debate, but yes, it has been taken into consideration by the US FDA, and it's just like there are so many research being done, as one being conducted in oncology, wherein the difference from 12 to 96% of several cancers through AI was observed. So you can understand that there's a lot of difference. So basically, it's not like you can just blindly believe the AI system, right? So the reproducibility issue that haunts the healthcare. So this is the buzzword wherein AI is facing, and that's the reason why we do have some guidelines being given by the World Health Organization and the FDA. So there are guidelines for implication of artificial intelligence system in healthcare. Fine. So even though there are some guidelines, you could see that FDA approval requires, and it debates a process that lacks equity, because FDA relies on having some benches discuss things. But you don't imagine African people sitting in the debate, right? So basically, it doesn't give equity because they are not even into the conversation. So that's what is the debate of the hour, wherein FDA, they say that FDA is not providing equity. So maybe it's a matter of debate for the future pharmacists. Right? So I just jump in 3D printing. 3D printing in healthcare allows to individualize medicine as per the patient needs. So, just for an example, oral disposable tablet instead of conventional tablets for non-compliant patients. Okay, so I suppose those who are PharmD and who are B.Pharm second year mode, they can understand what is non-compliant patient. So it's very difficult for us to give a drug. So if you are having a non-compliant patient, and you do have a disposable drug, it's easy for us to make more of compliance. Okay, so this manufacturing method seems to be especially beneficial in the production of orphan drug, and orphan drugs are future in USA. You must be knowing that through orphan drug you can minimize a lot of money in USA. So there's specific requirements for you to hit orphan drugs. It's like specialized medicine. So for you, in case you take your drug to be an orphan drug, it's like 20-30% of the revenue that you churn out will be more in your pocket. So many of the pharmaceutical companies concentrate more on designing their drug in terms of orphan drug that has been registered under the US FDA. They are relatively cost worthy, and as yesterday's chief guest was mentioning, there's a lot of impurities that have been there. But in 3D printing, that is 3D printing in healthcare, all these things will be minimized. So 3D printing has and eventually will become one of the most revolutionary and powerful tool, serving as a technology to precisely manufacture individually developed dosage forms, tissue engineering, and disease modeling. I'm happy to inform everyone that we do have a 3D printing machine in our university. Yes, we do have 3D printing wherein we print cell lines and tissues, if I'm not wrong. Yeah. but we don't have equipments to print medicines or tablets as of now. And with the help of individualization of treatment process, 3D technology will surely lay the foundation for treatment for less common diseases, and it's for sure. Right? So, as I had discussed earlier, 3D printing technology can surely bring down the drug discovery timeline to maybe 20% through its bioprinting concept prints. So this was earlier used during the COVID times, wherein there was a lot of RNA, tRNAs being designed and printed as a concept through bioprinting. And bioprinting nowadays is the buzzword in terms of manufacturing. And you can find a lot of COVID vaccines being produced through bioprinting, and you can browse through when you reach your home. So, as I mentioned earlier, it's more eco-friendly. Okay, the benefits of 3D technology in compounding pharmacy include eco-friendly nature. So there's very less of pollutants. Okay, and the ability to print small-scale drugs saves more of energy. You don't require a very big scale of manufacturing unit to be precise. The ability to replace a large number of drugs with one tablet. Say, suppose you are taking 4 or 5. I have diabetes. I've got some cardiac problem. I do have some psychiatric problem. You can give me a single pill that is 3D printed that comprises of all these drugs. Okay, so I've got those extra examples coming over. So this could be patient-centric, appropriate medications. Even during the pandemic, wherein only pharmacists and the healthcare would have worked drastically for the health of and life of many individuals. And for the manufacture of drugs, a 3D printer is the only piece of requirement. So, God you may have pharmacies in future having a 3D printer, meaning a patient can come speak to the pharmacist. They can say that I'm having this thing, that thing, through artificial intelligence, to telecommunication. There may be a doctor prescribing something, and they will print your drug immediately and give it to you. So this could happen. Science is so. Today's joke becomes tomorrow's reality. So maybe it's all possible. So these are some drugs. It's Aprecia Pharmaceuticals formulated an anti-epileptic medication that is levetiracetam, named Spetan. Okay, the first 3D printed drug. You guys okay? Maybe you can accompany them. Thank you. By the grace of God, I suppose no one was injured. Thank you. It's all blessings that keeps us alive. Okay. To continue with the topic, Aprecia Pharmaceuticals formulated that it's more of a reformulation. Okay, they reformulated epileptic medication called as levetiracetam, named Spetan. Okay, the first 3D printed tablet that disintegrates within seconds in an aqueous solution, wherein developed through a proprietary powder bed inject 3D printing technology known as zip dose. Similarly to a selective laser sintering SLS, the inject printing head drops a binding liquid onto a powdered layer. Okay, so there's a center. There are powdered layers that keep the layers intact. Depending on the size of the tablet, layers can be stacked up to 40 times, allowing for tighter packing of the drugs. In general, a single tablet containing 200 mg can contain 100 mg, and it was approved by the FDA in year 2015. So the resulting pill will be a high dose medicine that is easy for people to swallow. The drug breaks down in the body and is steadily absorbed. So this is one new of a 3D printed drug, which is called as polypill, designed and manufactured via 3D extrusion-based printing technology to treat diabetic patients who are also having hypertension. The polypill consists of a captopril osmotic pump compartment, joining layer, as I mentioned you earlier, and a sustained release compartment, nifedipine and glipizide. So as you may know, this captopril and nifedipine are for hypertension treatment, and glipizide is for the diabetic treatment. So these are single pills that work wonders. When taking this pill, the joining layer disintegrates quickly. Therefore, the polypill splits into a captopril compartment and sustained release compartment. 3D printing technology enables to regulate independent release of profiles controlling the porosity of diffusion. So this is just an example. So they are there are majority of drugs that require combination. So I believe 3D printing could be the future of pharmaceutics in future, where you have a lot of combination drugs with wonderful dispersion. So these is 3D printed personalized footwear with embedded sensors. So this is these are some buzzwords wherein you can print your own 3D printed footwear, and the data would be transferred to a cloud-based center through AI and augmented intelligence. You will also get those data being referred, and finally you will get a health diagnosis report directly sent to your doctor, and you will get all those things in your watch. When they may advise you when to take a breakfast, you're not walking straight, your gait is not proper. Things are fine. Augmented intelligence. We've been speaking about augmented artificial intelligence. Now it's more of augmented intelligence. Precisely, both are almost similar. However, while artificial intelligence enables machines to mimic human behavior, augmented intelligence uses the same machines but with a different perspective and goal in mind. Augmented intelligence brings machines and humans to enhance better healthcare. It's augmented intelligence is something that has cognition into it. Cognition refers to high level brain functionings like the ability to read, write, take decisions, and so on. Whereas artificial intelligence is just made to do things that are to be done. But augmented intelligence can think precisely, and it can judge, and it can suggest an artificial intelligence. So artificial intelligence with that of augmented intelligence can do wonders in the field of healthcare. So you can see here. So this is one of the example wherein the enhanced post-marketing surveillance that may allow a more streamlined pre-market review process of augmented intelligence algorithms and more frequent production modification over time. So you get this data very frequently, and this could impact a lot on the drug market cycle. So we've got the implementation of augmented intelligence in clinical practice too. Augmented intelligence, also known as intelligence amplification. So now you may understand. Augmented intelligence is amplification of your that is artificial intelligence. Okay, cognitive augmentation. So your cognition is being augmented through machines. The augmented intelligence helps human intelligence elements in procedure alive more machine human thinking. So it's more of you getting into kind of possessed with a machine that has been possessed with humans. That's how it is, and to simplify things. So this is something called as My Intelligent Machines. These are augmented intelligence softwares that capture and aggregates clinical experimental data which are structured and unstructured and help the users understand the patient heterogeneity. Heterogeneity, as you may we've got a lot of people in the pharma who suffer a lot because of heterogeneity, and that's a very costly issue to deal with. This allows scientists using My Intelligent Machine software to stratify large population of patients with heterogeneity who are dispersed geographically. In 2021, MIMS raised a whopping 8 million funding, and the expansion market such as is planned beyond 2023 for the coming to the future. So this is something called as Orbital RX. This is a hospital pharmacy solution, basically registered under the IBM Watson. They've been working for the past 20 years, IBM Watson, with artificial intelligence and augmented intelligence. So this is basically a drug supply chain management tool and evidence-based insights from micromedics. So it's basically looping micromedics, that's a drug information solution, supporting proactive and effective management of drug shortages. Eventually, you would have seen this a lot in terms of the pandemic that we have seen. Pharmacists were forced to work more sessions because of the lack of drug, right? Especially there was a period when one single drug was whooped and they had to manufacture those drugs in a large number. So these things the shortage of drugs could be brought down through use of Orbital RX. Orbital RX combines a real-time inventory. Okay, it's like what softwares you have in supermarkets kind, but it basically has the micromedics that gives the augmented intelligence to it. Right. So with the latest evidence-based treatment, the hospitals can consolidate, view procurement proportions, and all within a single click. That's how fast it is, because that's what machines are for. Okay, so this is something that is IBM Watson's registered. It's just a question I would like to throw to you, gentlemen, people, around IBM 4680 pharmacy system. So it's basically drug enforcement administration device kind. So it's more of a robotic stuff. Any guess when it would have been launched? Any guess? Anyone can please? Not for those who have seen my slides. You can see, sorry, 1989. So this is the announcement letter given by the FDA. It's 1989. Okay. So basically, the IBM 4680 pharmacy system helps automate traditional and unit dose dispensing in hospitals, nursing homes, retail drugstores also. The system produces drug information administration reports, patient profiles, and cross-margin reports. The IBM 4680 pharmacy system provides an online update of patients, doctor files, drug interaction, allergic reactions, and so on and so. You may feel that we are being overwhelmed, but it's not reached India also. For pharmacists in India, they not worry about it. So this is something called as robotic process automation, RPA. The critical difference is that RPA is a process driven, whereas AI is data driven. So process driven and AI, more of data driven. Robotic process automation, also known as software robotics, uses automation technology to mimic back office tasks, like what you do in your office, right? Normal tasks like fill forms and all those things. You move files across day-to-day basis, taking ledgering, data filing, and all those things. This form of automation uses rule-based software to perform activities at a high volume, meaning a lot of volume, especially that are evident with that of the insurance people. They require a lot of data to be embarked on. It is also more common with that of the medical stuff also. So especially these are used in claim processing, that is processing health insurance data, as I mentioned earlier, payment processing in health insurance, and appointment scheduling, compliance management, discharge audits, medical audits, pharma audits, whatever you can term as, medical legal services. Yes, I saw an article, this was into medical legal services. And you can put in a mail drugs of choice and your with the prescription of a physician. Yes. These are digital surgeries. Eventually, this is for people who don't really believe in work. But if, say for instance, if some we've got something called a cyber knife, yeah, cyber knife. So when they ask whether you want a cyber knife or a human surgeon to do it, everyone prefers cyber knife. So that's the confidence that we have on robotics because of the precision. So digital surgery is use of technology for enhancement of pre-operative planning, surgical performance, therapeutic support, training, and to improve outcomes and to reduce the harm that surgical procedures may give. Even bleeding and all those things also will be very minimal when you use digital surgery. So I'm not advertising anything here. It's just for your information. That's it. So I'm not being paid by this Da Vinci system anyway. More than 60,000 surgeons around the world have been trained on Da Vinci systems and have completed more than 10 million surgical procedures using the Da Vinci system. The Da Vinci system delivers 3D high definition views, giving surgeons a crystal clear view that is amplified 10 times that a normal eye cannot see. Okay, so the surgeons use instruments that are more likely with less of vibration, which are very far greater and precise than that of the normal things that a human uses. Okay. So we've seen a lot of discussion on the big data analytics, but I will be more precisely speaking about big data analytics in the pharmacy setting. The term big data is commonly used to refer data sheets that are too large and heterogeneous for humans to, invade into. Data scientists usually integrate the artificial intelligence-powered analytics to, constructively evaluate things. So in hospital and healthcare settings, big data sets results from the documentation of pharmacy interventions, medication reconciliations, and patient monitoring. So this all requires a lot of data, and when you bring this data into big data analytics, we have a lot of data being being pulled into as a direct result of these activities. Patient electronic health records, even including medication history, adverse drug reactions, pharmacokinetic consultation, all those things, they could be brought into fingertips and could be given to your mobile app also. So I think you may be, this is the one thing that more speakers didn't pronounce, so I'm happy to speak about it. It's called as smart algorithms, and these are okay. You're not allowing me to use this? Are you allowing 636? Okay. Now my technology is so always over this. So smart algorithms, these are complex algorithms which will soon help clinicians make incredible accurate determination about the health of large amounts of information they're premised large expandable correlation of data. Okay, so in 2014, Alive course algorithm for detection of atrial fibrillation was approved by the US FDA. It was approved by the US FDA, and 2 years later, US FDA found further 4 solutions ready to clinical use, while in 2017, 6 new algorithms were approved by the US regulator. So this exponential growth accelerated in the recent years, and when US FDA had endorsed 23 algorithms, and I believe it's not going to go down. For instance, there was one algorithm designed by Google that used data sets on eye scans from over 125,000 patients to build an algorithm that could detect retinopathy, that is number one cause of blindness in the globe. So they had designed this algorithm wherein 90% of accuracy on par with that of the board certified ophthalmologist was observed. So again, these results had some constraints, but as such, as first approvals in 2019, they're never turning back. There's a lot of algorithms being marketed. You can see more of algorithms in terms of dietetics, where you could get your personalized dietetics based on algorithms. You could see in TEDx a lot of algorithms being released, wherein they set different algorithms for diabetic patients, and they do suggest a specific carbohydrate intake and all those things with precise algorithms that work precisely for a genetic makeover. So as I mentioned, big data is expected to penetrate faster and deeper into the healthcare sector than in other industries, like for instance of media, financial and manufacturing, etc. So to answer this question, what does the future hold for? I believe younger generation would have gathered to listen to these things. Something called as meta ecosystem. Okay, it's basically an ecosystem wherein you involve virtual, you involve the raw data, that is allow control open access, tomographic raw data, etc. You also use augmented regulatory science. It is called as augmented regulatory science to extend virtual clinical trials. So there's a virtual clinical trial that are set to start in terms of scope of duration, and the metaverse medical intervention will perform medical intervention aimed by the metaverse. So these are some ecosystems that are possible to hit the market. Next is the mutation burden. This is something that has very less work been done. We've got what do you call it as antibiotic resistance stuff, but this is something called as a mutation burden, wherein industry people will be knowing because they work with a visionary before 15 years and they know better. So there's something called as a mutation burden, wherein one drug that works with you may not work with me. So this may be the future where clinical pharmacists precisely will have to work very enormously with healthcare system. So there are something called as closed loop apps. Okay, there are closed loop delivery systems. I'll just give one example. There are a lot in market. I think in market or in trials. There's one called as closed loop app CamMax. Okay, it's CamDiab involves. It was designed by Cambridge University. That is an adaptive model predicts control algorithms. So algorithms, okay, with software and Android smartphone which receives data in continuous glucose monitoring. That is called as Dexon, and directs insulin delivery via insulin pen. Okay, it's insulin pump that is called as Dana Diab Care. The closed loop app it is initiated using a participant's total daily insulin dose and body weight. So it calculates all those things. Every 8 to 12 minutes, it responds by giving you the appropriate dose depending on your requirements. So the algorithm keeps developing. It starts giving the data to the AI, the cloud-based system, and finally the AI algorithm is on daily basis being revamped specifically for you. So that's how closed-loop delivery systems would be. You can have one in your body and just start doing your work, and you're not worry for 2-3 days, hopefully. So this is what I was mentioning. Campex, this was by the Cambridge University. So these are wearable full body motion trackers. So basically, this would do wonders. It's not for people who are not able to walk and all those things. It could be given to me also, given by my wife to track myself to provide a. So these are things wherein, if say suppose you think that I am susceptible to a cardiac arrest, you could ask me to wear this, and there'll be someone who will be helping immediately, because we've got satellites, we've got Apple working with direct satellite system SOS. So you connect that thing without internet requirement, direct satellite linking, you can save me down. So that's how fast things are. And yesterday, as Dr. Rao had pronounced, it's more of genomics, genetic stuff that would tailor the precision medicine. So precision medicine is here, it is in, and it will be there for the future to come. And something called as next generation sequencing, that is the NGS test. These are basically would be done before we will be given drugs. So you'll be having a set of NGS tests. You'll be having a card time when you visit the doctor, and these data will be given before you've been prescribed, and these will have your mutation data, each and everything, your algorithms and everything. So this will be the future. Okay, so even in terms of evidence-based medicine, as yesterday you have seen, Dr. Rao mentioned, the pyramid has slowly become an iceberg. So evidence-based medicines, yes, will be given to the doctor's fraternity for better patient care, where they just need to key in, like what we saw the chatbot of the Chinese chatbot. We just need to key in things, and all these guidelines will be evidence-based, and you get fingertips of the diagnosis that is precise and tailored specifically for the patient. In conclusion, I would like to conclude, as with any technology, there will be challenges associated with incorporating technology into traditional healthcare. But implementation effective, that is, but if it is implemented effectively, the benefits are likely to massively outweigh the risks. I request the whole pharmacy fraternity to embrace the changes from mere compounding to patient care, as mentioned by our guest of honor yesterday from TCS. So even technology-oriented people, they see, they are at watch with the pharmacy professionals. With this, I would like to end my session. I've given my phone number and email ID. If in case you require all these are all articles taken from open source. If you require these articles, you could please ping me in any means, and I will be happy to share all the articles for you. Thank you. I'd be happy to take up with this.
This is the next presentation.
Thank you. Thank you again. Again, I would like to take this opportunity in extending my gratitude to the organizers again, Dr. Prakash Gowdanava, Dr. Ramesh, my mentor and dean, to have considered me worthy of standing and sharing this podium with giants in the field. Thank you so much, sir. Thank you.
Thank you, sir, for sharing your precious ideas and thoughts.
I request Dr. B. Ramesh to felicitate Dr. Rajeshwi, and I request Dr. Prakash Gowdanava to join the felicitation.
Thank you, sir. Now we have with us Mr. Raja Agnihotri, Chief Strategy Officer, Advani Research Private Limited, Clinical Research Organization, Hyderabad, India, for the second keynote session.
Now started already.
Sir is here to educate us on growth strategies for pharmaceutical industries. I request Mr. Raja Agnihotri to take over.
I can handle there, I will handle. Pointer is there. I can do anything. Put the first
Same pointer I can give.
Which one? Right one.
So this is the next one. This one is next. This is old.
No, I have an image. So do I press this? It will
Ah,
come.
yes, sir. Yes, sir. And I'll be here, sir. If you want.
Color logo. Color में भी.
Yes.
इतना कुछ कर दो color में.
सर जी वो ऊपर से लेके आऊँ
Check the time. To show. Slow become slow.
Hello. Good morning to you all. It's a great pleasure to meet you all. In fact, Dr. Rajesh's presentations made my going forward a bit easy. I'm not sure how many of you could get it, but that's how the industry is working now. And with his slides, don't get too much bogged up, and you will always get confused that where are we in the pharmacy or are we in the digital world, right? But that's how it is going to be. That healthcare is going to be in the future. There will be complete integration of science. There will be complete integration of digital and the patient needs. All is going to be integrated. In fact, many of the global projects I'm being involved currently, and I'm executing many of these projects, so I know how fast these things are going. So looking at these slides, don't ever get confused that did we choose the profession correctly? Should I be in IT or something else? You're all going to be called as data scientists going forward, right? You will be definitely called as a data lead or a data scientist going forward, because you deal with so much amount of data, and there will be science which you would be your core domain expertise. And with that core domain expertise and with available data, that's going to be the advancement in the pharmaceutical industry. This is how it is going to be. And please take my words a bit seriously that if you don't want to stay there, nobody is going to wait for you. It will go. Science is going forward. Pharma will go forward. And if you are not keeping yourself updated in many of these things, yeah, you will be there where you are. Right? And it's all a good opportunity for you all, because you will start your career now reading this. Right? Please pardon us for that. Why? Because at this stage we have to read around 17 to 18 hours per day. Because to get this, to know all this, we come from a core, what I can say, pharma, which was 20-30 years back. We started our career from that perspective. Now this technology has come and we need to integrate. And when we integrate, those challenges are always there. And we have to start from the basics, and we need to gear up for all this. So it's a very, very important for you all that you have to be on the same page, not only in the science, but also in the technology and the integration, how it is brought into this platform. Very, very crucial. Right? And some of his slides were very mind-boggling, and he could capture good things and made my presentation a bit easy. I am not sure how many of you could catch that robotic surgery. In fact, for the first installment which happened in Apollo hospitals in Delhi, I was one of the team, because we did a clinical trial in that robotic surgery. It is from an intuitive company from US. Right. And the way it is done is completely data is handled around 40 kilometers away from the site of operation. Nobody is there. So we took around 45 days of training, and they took training from us to understand the post-operative clinical procedures and how many data we could collect from that. It will be mind-boggling that from our side itself, where we were a part of a vendor for that, we gave around 6,343 data points to be collected. And those data we collated, and it was useful for them in the part of how do we use it in a post-operative surgery. As of now, it is being used for gastrointestinal surgery. They are now moving forward towards cardiovascular surgery also. Intuitive is the company. Please keep an eye on that. And Da Vinci is the brand.
Video. Hello. it's on.
Yeah, again we are back. So I left that robotic surgery. Don't ever think that it is an equipment in what way we are concerned to that, but there is a lot of pharmaceutical inclination towards that equipment, and they manufacture only 6 or 7 like that. And India is now having 2 installations. One is in Kolkata, another is in Delhi, and now the third one is getting installed in Apollo, Bangalore. So this is going to increase, and when it is going to increase, 100% you all will have more opportunity to get associated with such kind of job opportunities to work and deal with those kind of equipments. Now, coming to this, I think you all know, I think there are some online also. I say hello to them also. The topic was very interesting, and I thank the organizers as well as the college, and a great opportunity for the students. Why? Because international conference is one thing, innovation and advancements in pharmaceutical sciences. Innovation is one, and advances is the other. There are many things which are happening in pharmaceutical sciences. I can say it was moving around 30 kilometers per hour speed before pandemic. Now we are moving close to 600 kilometers per hour speed. That's the kind of speed we are working now in pharmaceutical industry. So that makes me interesting. What I have done is that my journey in this pharmaceutical industry has been 3 decades. I'm entering in the 4th decade now. I've been in bulk drugs, API, branded, and equipment now for the past 2.5 decades in clinical research, starting from drug discovery, regulatory, involved in great launches in India as well as globally, traveled across the globe and in many panels on the policy decision making, and continuously contributing towards the clinical research industry as well as with the biosimilars or biotech therapeutics, whatever we want to call it. And that makes me interesting to see this. And very importantly, you all should know is that the topic is very, very relevant to what has been the speakers yesterday and the today. And yesterday I could see, though I couldn't be a part of it, there was genomics which was covered, there was novel drug delivery systems which was being covered, and today the integration of the science with the technology you are seeing. So what I'm going to talk is all my experience, which I'm going to share with you all. Since I represent a clinical research organization, the contents and the talk, what I do is all on my own, nothing related to the organization. They have at least given me a permission to talk. So all the contents have been picked up from my experience, and the thoughts and the discussions, what I'm going to do is all my interpretations of my journey for this 3 decades, and I'm going to keep you engaged in all these things. And wherever you feel that you have some more clarity required, please write it down at the end of the session. I think we can debate or talk, and as far as my knowledge goes, I'll try to see that if I can give a suitable answer for that. The topic what I am growth strategies for the pharmaceutical industry. And it's a very wide topic. Pharmaceutical industry is very, very big because it has been now segmented, fragmented into many, many parts. So I'll touch base on important things. What I've done is that since the audience is more of students and career starting, I'll try to give a glimpse of opportunities where you can be in that. And interestingly is that I have taken growth strategies, And growth in the pharmaceutical industry has happened in three directions. One is, we call this as a direct growth from the organic, means each and every industry will have a direct growth with their portfolio of products and operations. And the second one is that the industry itself has expanded vertically as well as horizontally. Horizontally in terms of allied sectors which are associated with the industry. And another aspect is acquisitions which are happening very rapidly all across. And importantly is that the Indian companies are playing a major role in this acquisition. So all types of things puts into a growth. And whenever you think or read or see something is happening, it doesn't happen overnight. The work has been started or would have been started maybe a year back. A lot of thought process goes into that, and there is always a strategy. So the growth doesn't come just like that. There is a strategy like that. Only one thing which happens without a strategy is this age. It keeps on growing. Without any strategy, you keep on growing. Gray hairs keep on coming. It doesn't require any strategy for that. But for the industry, yes, there are some strategies. When I say the word growth, first and foremost is that it is positive. Yes or no? Nobody would like to be saying that degrown. I have degrown or the industry has degrown. The word growth itself is a very, very positive for you all. Yes or no? He has grown from last year. He has grown from last 2 years back, and now I'm saying it has grown. So the word growth itself is very, very positive for you all. So rest be assured that you are in an industry which is growing. Don't ever think that it is degrowing. It is growing. It is very, very positive that you are in the right industry. And second thing is that whenever there is a growth, there is always an eyes on it. Means more focused on it. Now, after this pandemic and during the pandemic, because of vaccines, people started looking at us in a different binoculars or a different lens, and everybody was more attentive to understand what is this pharmacy is, what is this pharmaceutical is, what is this vaccine is, what is this clinical trial is. So many things came into that. And whenever there is a growth, there is always an eyes on it. Means you are in the observation scanner all the time. Got my point? So now the pharmaceutical industry is that, because it is been growing in such a fast way that everybody eyes are on that. And secondly, whenever there is a growth, whenever there is a keen observation, it becomes a point of measure. Today I have grown, tomorrow they say, okay, they expect at least growth over that particular base. If there is a degrowth, then they will say, from the last year you have not grown. Same thing happens, I think, in the marks also. If I am right, please don't change. If at all you want to change, let me know.
1 minute, sir.
Why do you want to meddle with this? Why do you want to touch it? I think you run it. You want to do presentations on your own?
No, sir.
Then why do you want to show it?
Yes, sir.
Don't even touch it, please.
Okay, sorry, sir.
There is a point of measure. Point of measure means always when there is a growth, they will look at from this base how the performances goes forward. And industry as such also, when there is a growth, it is always measured. Last year to this year, last quarter to this quarter, last 3 years back, last 4 years back, last 5 years back. Why this becomes an important? You can ask me a question that why should we see so closely? Maybe not important at this point of your life journey, but as you enter into that, into the industry, the fundings which happen, or I can say the expansions which happens in the industry is always made, or the decisions made to grow or to enter into a new market with this point of measure of growth. Okay, 3 years this organization is consistently growing, let me invest. This segment is consistently growing, let me see if I can launch new products. This segment is multiply growing in this geography, let me plan and strategize if I can go and enter my product or launch my product into that geography. That's why it becomes a point of measure when you say growth there, right? And I'm not relating just that pharmaceutical. Now, when we come to the word pharmaceutical, it is very important for us to see related to health. I'm using this word in my coming presentations. You will know why I use the word health. All activities, whatever it is, we have seen yesterday genomics, novel drug delivery systems, or today morning Dr. Rajesh presentation of AI or precision medicine. What it's going to do ultimately? It has to improve or enhance the quality of life, which we holistically start speaking now of health. There is a radical change in the concepts of the product, concepts in the pharmaceutical industry was, I'm bringing there, I'm stressing on there, please think very seriously on that. In my 3 decades of journey, whenever we used to launch a product, there used to be a disease manifestations already there. And we launch a product to control that disease so that it doesn't spread. Disease happened, you launch a product to control it. But now we are coming out with the tools to see that disease management and prevention. And how do you maintain it so that your quality of life improves? That's why holistically the word health comes whenever there is a pharmaceutical brand name or a generic name whenever we use it. So we are directly responsible for the health. Whatever you do, ultimately it goes to the improvement on the increasing or enhancing the quality of life. That's why the word health is. And second thing, as I explained, is the disease management and sometimes cure. There are many segments we need to manage the diseases, and there are very steps innovatively done to cure and prevent it so that there is no recurrence. So these are the things which are happening in pharmaceutical industry. That's what my point was. And very interestingly is it's evolving. I cannot say month or a year. I am saying by minute it's evolving. It's evolving by minute. Each and every one is coming out with a new concept. Each and every one is coming out with a different innovative approach to control the diseases growth, unknowing diseases, untapped diseases, untoward incidents. All those things are so much happening, and that's why even in the industry also it is evolving. You got my point. So all these points lead us to have a proper strategy from the industry perspective. Please, it's a very, sometimes I feel myself that it's sad, but we are all in this business. For this industry to grow, there should be a disease, there should be a patient. Very unfortunate, right? If we say of health, we say of many things, but for this industry, suppose if I say my industry is growing, it means that so many diseases are there, so much of people is consuming the medicine. So for the industry to grow, there will be this kind of parallel activities which happens, and it's very evolving. From these slides, you can take 2 ideas. One is that you are in the right industry where there is growth, and you are in the right industry Why? Because you are concerned directly to the health of the population. And the third is it is evolving. It doesn't stop. Right? It is evolving. Means more opportunity for you. Now, this I think you will get it everywhere. As I said, it's evolving. I'm just touching this slide on global pharmaceutical industry as that. It is around 1,136.23 billion as of 2022 and 5.6 as usual. North America is more contributing towards the pharmaceutical industry. Asia-Pac has picked up, and India in terms of exports has picked up, and India's contribution to the global health has increased tremendously with the launch of this COVID vaccines and also many generics which are entering into the regulated market like US and Europe. And I think you all know about this IBEF. If you don't know, please Google it. Now, Ministry of Health and Chemicals and Fertilizers, every year they release a report, very consolidated report. And that report, it is a PDF, it is publicly available. You can download that report. It starts from very basic things. And it will go till how many drugs are launched, how many imports are there, exports are there, which country. So it will also give you an idea of what's happening in the industry. That's why I'm not going deep into that. This will give domestic market. Of course, it is around $42 billion. If I have to convert that into Indian thing, as per the last month, it was close to 2 lakh crores rupees. At a monthly around 20,000 to 23,000 crores per month on a moving annual total, we call it as. And to be very precise, when I started my career, there was in this pharmaceutical industry, there was a small segment around 12 cardiovascular, respiratory, ortho like that. But today we have got 64 segments in that. And these segments will give you an idea that how it is being fragmented, how focused the industry is growing. Right? This is all available in public domain. You can see it. And this will give you an information that we are in a growing and evolving industry. More than that, now this is very important. That's why even 2 days back, I was talking to Dr. Prakash. When you say about pharmaceutical industry, what point you want me to talk? Why? Because it is very wide. Pharmaceutical industry covers generic drugs. Again, in these generics, we have sub-classifications like simple generics, complex generics, small molecules, large molecules, that kind of segmentation has started. Then we call OTC or the over-the-counter medicines which are being taken, consumed without the prescriptions of a physician. So they are all called as OTC medicines. Then we have API and bulk. API, all must be knowing, it is active pharmaceutical ingredient, and the bulk and intermediates is also there. And that intermediates includes your excipients, binders, dilutants. That's also a big market. Then comes vaccines. In this vaccines, again, there are sub-classifications like viral cell vaccine, mammalian cells, biotherapeutics, so many types of segments are there. Then comes contract research and manufacturing, where the formulations is, I have an idea, I outsource it to somebody, they develop it because they have a facility for that, and they manufacture and give. That's what we call it as contract research. And biosimilars and biologics. It is again a biotech process involved product. These were the segments in the pharmaceutical industry which were there earlier also, and some segments has been added. Now, apart from this, there is a huge amount of allied sectors which are also being covered in pharmaceutical industry. For example, equipments. Right? There is a huge market for equipments which are being used in the manufacture of pharmaceuticals, API as well as bulk. Then comes your supply chain. I'm not sure how many of you are aware of it. We call that as a distribution channels. And there is quite a number of innovative models which are coming out in distribution, because most of these products you would have seen on the label or on a label claim should be stored between 2 to 3, sometimes -2. If it is a blood product, it has to go into the freezing technologies like that. So that's again a huge segment which is there. That is also there. And also the industry which has gained more prominence and picking up in the pharmaceutical is packaging and labeling. Right? He touched about precision medicine. This is going to be the future. So all individual medicines will be packed individually to the patient. Individually to the patient. That's also we are now doing it in a clinical trial called as DCT trial, decentralized clinical trial, where we ensure that it goes directly manufactured to the patient as required, and it's being delivered to the patient directly. And all these methods were there, but it took more prominence during the pandemic because of lockdown, movements were restricted and all. Innovative ideas have come. So those are the other things which has been put into the pharmaceutical industry. So it's a huge, I'm telling you. This also gives you an idea that when you finish your graduations in pharmacy or a full grad, don't ever think that I have to go to manufacturing and all. That is huge. So all these things will come. Depending on your individual capability and capacity, you can tap where you want to go. These are them. I have just touched something. Now, all these medicines, when I talk about growth strategies in pharmaceutical industry, any industry, what do? They study the market. So either you can take it this or there, you found the market to grow, or you have a market, you back calculated and see what you can feed to the market. Both words are there. Importantly, for the industry to grow, there should be a market first. Correct? Without the market, where do you want to sell it and how do you want to get it? So the market becomes very important for all these industries to grow, for all these things to evolve. So market is important to understand. Then product portfolio. This is the first growth strategies they use. Which product, which segment they should be in. This is the first basic work they do. I cannot be in all segments. I cannot go into all geography. So the portfolio of products becomes very, very important. So they select the market, they select the product portfolio and plan their growth. This is how the industry was earlier. Or I manufacture a product, search a market, develop a strategy to grow. This is the first point which happens. So identifying the product portfolio is very, very crucial. It also gives you an idea that I think you all must be having pharmacology as a subject, correct? And there will be pharmaceutical chemistry, pharmaceutics, like that. Not all students will be good in all subjects. Some may be good in chemistry, some may be good in physics and all. So you select depending on your interest and your strength, the portfolio where you want to build your career. The same thing happens here. Next is reach. Our strategies of the growth is always dependent first on the product, right? Second is the reach. Reach here, how fast I can go to the end consumer, or how do I go? Very, very important. And which geography I go, that is where the strategy starts. If you be in a stagnant market, growth will not be there. If at all you have to go, then you should have a good reach. Then we plan everything. So the reach becomes very, very important as a strategic point of view in terms of growth. That's where supply chain, distribution, packaging and labeling, all factors come in that reach. Suppose I have a product which is highly unstable after 24 hours, but I have to send it to US. I have my market is that I have to reach. So the whole strategy revolves around how to go to US with this unstable product. So I develop strategies first to prevent the stability, have a consistent stability, develop a packaging, develop a labeling for that, and how do I ship it to maintain the stability. So all strategies revolve around that. So who does that effectively? There will be a growth in that. That's how the reach is very, very important. So most of us don't think that do we have an opportunity in distribution and supply chain. We play a major role there. Then segment. As I said that when the industry started, there used to be 13, 14 segments. Now we are touching close to 64 segments. So any growth which they plan or strategize, they will never think that I should be spread across all the segments. I want to be in antibiotics, I want to be in respiratory, I want to be in derma, I want to be in cardiovascular, I want to be in rheumatology. No, it will not happen like that. Either they select a portfolio which will cater to the different segments, or I try to consolidate in a single segment. How do I want to do it? So understanding of the disease segment and the wider the segments I can cover will help in the growth of the individual organization. And of course, now is not that point, but still we do keep it in mind whenever we develop a strategies affordability. Right? The end user should be affordable. When we were working around 1.5 decades back, the first point used to be questioned is, you do so many things, what is the cost of the treatment? We call it as cost of the treatment. And since many of these lifestyle diseases has to be taken for a life, it is a recurrent expenses for the patient. And how is he going to earn? And that earn can be reimbursement from the insurance, medical claim, or individual earning. I'm just giving you an idea that how we work. When we go into individual earning, we keep the per capita income of individual in the geography where we are operating. Do you all know what is the per capita income in India? Please keep that in mind. If the per capita income is less than 10,000 rupees and your own treatment, what product you are going to launch is going to cost 6,000 rupees, he is not going to buy. Is he going to buy? He is not going to buy. And this is where the whole planning and the strategy starts. This is where you will have to invent in such a way, or the pathway what you select, you need to do in such a way that the per capita income is what? And how much subsidies he is getting in health, how much the government is supporting in that. What is his growth opportunity? See, many a times I have to pitch to many of the investors telling that this is the growth. And the growth is coming from opportunities. Like I should not use the word target audience. For you all, the audiences who use the medicine will be in a wider sector. Need not be from a high income group. So how do we plan it? So that's where the affordability comes. You cannot make a product which is so expensive. Today, why there are more deaths in oncology, in cancer, why there is more deaths? Affordability is not there. The cost of the medicine, the cost of the treatment is so high. People stop after, or they may not go even for one cycle of chemo. That's why the whole research or everybody is putting energy into this area, oncology. Why? Because to bring the cost of therapy down so that everybody can afford for it. So that we can control or manage the disease or cure the disease. So much of research is happening in oncology, which in further slides I will tell you. Why the new dosage forms come, this is because of that. And today, you have to correlate like that. That is how the strategies are made. Suppose I launch a product in some parts of New Zealand, suppose for example, I look into total demography, income, government schemes, all kinds of things. Which sector is playing their major role, Whether IT is there, infrastructure is there, buildings are there, how do they earn their money, what is their lifestyle, what do they eat, what do they eat for in the morning, breakfast, lunch, dinner, then how the diseases can come, how do they manage it. So you have to know everything to plan a product portfolio. You got my point. So the health is related to so many things. So the growth will happen in that way only. So please tune your mind in that way. Then you will be very, very, I can say, prominent or successful. So these are the important things. You select this, you have a market, then you have the segments in the industry, then the strategies are being planned for that. But the basic thing for all of this is this. This is how a man visualizes. See, I have put this picture with a lot of logic into it. It doesn't have anything. What is seen, he will see that, he will build it up, he will find a path to get a solution. Now I'm going into the current structure and some of the strategies which are being used by the pharmaceutical industry. Core thing for anyone is research and development. This is the first seed in a strategy of a growth. Of course, in many organizations, either the R&D will give a suggestion or the top think tank for the business development or a business analytics guys, what we call now, they give that this is growing, this segment is important, why don't we bring a product there? Then the R&D starts developing it, they take it, they do research and they develop it. There are two different things. Always we use R&D as a single. First thing is the research. They find, then they develop it. Two things happen there. Right? So this becomes the first step of a strategy for a growth. If the R&D is strong, right, then definitely there will be growth. Usually in the pharmaceutical industry, many of the pharmaceutical companies, if you look at the annual report, suppose you all maybe 30% or 40% would like to go into R&D sector. So how do you know that the R&D sector is doing well? All of these listed companies, they have their annual report in public domain. You take any public listed company. In India, we have Biocon, like that, their subsidiaries like Syngene and all, they are all public limited companies. Reports will be available in the public domain. You go there, see the annual report. Thing is that you should have patience to read for that. In that, there will be written what is the revenue. And if you look at that, more than 30% of the revenue they allocate for R&D budgets. If somebody is earning 100 rupees, 30 rupees is pumped back into the R&D. Some may 10%, some may 20%, some may 30%. So it depends. The more they spend, is always a forward-thinking organization who wants to spend and research and develop it. So that gives you an idea that they are more R&D focused. I have an opportunity if I am innovative, if I am able to think in a different way. That's how you have to visualize and reconnect. So R&D becomes a very important strategy for the growth. Now, dosage forms, I'm just writing it, I'm just touching it superficially. If we look at earlier, we used to have a tablet, a capsule, a syrup, or a tongue, maximum. That used to be, and still many companies are into that space. Now, slowly, ointments came. Slowly, creams came. Why do you think that ointment and creams have come? Just because they have so much of water in the facility, they started. It's not like that. It is, they will always think at the end user convenience. Suppose you have a joint pain and you want to give 3 more tablets inside it to avoid that extra ingestion of the tablets. If something on a surface, if you can apply or do it, it can act locally. That's how the new dosage forms have come. And today, you look at it, I'm sure you would all know that multi-layer tablet, sustained release tablet, bi-layer tablet, extended modified release tablet, pellets, granules, so many things have come. Why do these dosage forms come? That is the strategy. They want to create a niche. Right? And delivery platforms. This is a very, very growing. It is a very short of time for me to explain, but I can tell transdermal patches, in that granules have come, depot injections have come, nasal sprays have come, inhalers have come, powders have come. So many things have come. And all of this has enhanced the patient experience with the drug or with the disease. Ease of application is always kept, and at the same time safety and efficacy. So from a simple solid dosage form, people have moved, and this has moved only with the pharmaceutical industry, only with the brain of the pharmacist like you are. Today in every board, every activity, microencapsulation is there. And they have gone to such an advanced that targeted delivery systems. People are working. If you can, I don't know how seriously you are looking at this slide. Depots direct. Protein bindings are there, which will direct to the site of action and do it. We are doing 2 trials on that, especially in gastrointestinal tract. We would direct to the protein absorption, see that the drug is released at that particular site of action. So many things have happened, and that becomes a strategy for the growth. And so much has evolved in this dosage forms and delivery platforms, right? And even in injections, single injections, point injection, needless injections, painless injections, and meter dose injections. Insulin and all used to be a big tube earlier. Now you get a single dose insulin injector. You don't even feel the pain. This is all because of delivery platform. That comes to after the R&D, they go into the manufacturing. Again, facility. Earlier, the path of a facility used to be that I will have a single punch machine or a double punch machine, which will give me 100 tablets per day or 100 capsules per day, and all those things. Today, the facility is done keeping long term as well as short term. What are, if I want to take this product to FDA, that US FDA, I mean, so they keep the US FDA norms and they build the facility in such a way so that the product manufactured here, I can take it up. And that's why the GMP part of it, which you would all read it, has also enhanced. So that becomes again a strategy for that. And secondly is the equipment. We have seen equipment very manually, means completely manually driven. Then came semi-automatic. Now there are 80% of the equipment which have gone into fully automatic. Means you need not have to be there. You can feed it to run on its own. That's where now in this slide also you were saying about collecting the data, collecting and putting it. AI or machine learning is playing an important role there. So when you say manufacturing, please don't look at only manufacturing a product. It is also the facility. It is also the equipments which are being involved in the developing the strategy. Now, second important strategy which everyone works is the product portfolio. Which product I should launch? Which segment I should launch? There are organizations which focus on only one segment and they want to be leader into that. We call that as niche and segment leadership. Means I want to be in derma, I want to be called a leader in a derma. So they keep on innovating products in that particular segment. From 1 to 100, they look into all those. So that becomes one of the strategies. They think that which segment is good, which segment is potential, which segment I should launch the product. Accordingly, they will bring the product portfolio and also think on segment profiling. Again, once this is done, they go back to the R&D. If the product is not there, then they come to the manufacturing, build up facilities and equipments to meet this segment leadership and plan the growth. The third, of course, is very prominent nowadays is the regulatory pathway. Regulatory pathway becomes very important because you should know, if you recollect in my previous slide, I said that reach. Earlier, most of the Indian companies, the pathway was first domestic market, then they go into ROW market. We call that as the rest of the world market, and then they would slowly enter into Middle East and North Africa, or sometimes I can say GCC, Gulf compliance countries. So then they would go into USFDA or EMA like that. But now people are so geared up, talent is so geared up, the facilities and equipment in the beginning itself, they plan that I should hit USFDA. And you all know that it is Indian companies which is having maximum filings, ANDA filings we call it as abbreviated new drug application, maximum per year in US. And apart from US, it is in India where there is maximum FDA approved manufacturing facilities there. So that's where the regulatory pathway helps. How do you go? Accordingly, they build up the portfolio, build up the facility for that. And this is mainly done to penetrate the market and consolidate the market. I want to go to USFDA first and penetrate there, then I choose which product, which portfolio, which dosage form. Then I launch them. You look at any generic drug, it will be always Indian companies who have launched there in most of the regulated as well as semi and non-regulated market, because pakka understanding of the regulatory pathway. And this plays a very important role. Even in the clinical trials also, we plan the clinical trials in such a way, understanding the regulatory requirements, and helps us in launch of the product through that markets quite effective. And lastly, I said supply chain and packaging. If all these things are there, you don't have an effective supply chain and packaging, you will be nowhere. So that becomes a lot of money is being pumped in. Close to 10% of our revenue earned, we are now pushing into supply chain and packaging. If you look, that's where you should keep on looking at the packaging of any brand or a medicine you take it. Alu pack and all, they will be marketing. I am sure you will be aware of it. These are all coming, keeping in the physical as well as incompatibility characteristics of the product. The packaging is recommended. Earlier, it used to be more of an aluminium and a crystal ply. Now you see how many individual packagings and supply chains we do it. That's all because stability is one, reach is one, and I think to meet the unmet needs of the end user. Means end user, how does he consume it? Keeping that in mind, these things are there. So this covers basically all the strategies which are being involved in most of the pharma companies for their growth. Apart from this, and this as I was explaining, it's all interconnected and everything. It is not only that I do R&D, I don't take care of supply chain, or I don't take care of manufacturing. It doesn't work like that. It has to be completely integrated in the product chain or a life cycle chain. But Rajesh's slides made me very easy. This has been challenged for us from during pandemic and post-pandemic. Healthcare, now the word healthcare I'm using. Why? Because the whole patient needs, disease, manufacturer, or the research, or the supply chain, they are all coming in a one word, which is called how do we improve the wellness of the public. The word wellness is coming. And with this, unfortunately or fortunately, the technology has entered because of COVID. Now, when it has come, people have seen how it will meet their requirements. The healthcare itself has completely defined in a different way. This is going to be the new healthcare. That's why I say, welcome to the new world of healthcare. Means I manufacture, somebody gives you, you take a pill. It's not going to be like that. It's definitely not going to be like that. The consumer is at the hand. Consumer means who is consumer, he becomes the boss. He decides what I should take. And he will be the success or a growth for the product or a company. Today, because of internet, before you go to a physician, you would have read many things, and you yourself will be behaving like a doctor in front of a doctor. No, sir, that medicine didn't work, this medicine didn't work, this Google is saying this, this literature is saying this. So the consumer has become like that. So every manufacturer is thinking in a different way now. Those who go beyond this traditional one-off interactions of yesterday, on-demand health experience, this is even we are facing it. Day in and day out, we are facing it. On-demand, I have paid you money, please give me that. They want to experience it, like a movie. When you go to a movie, you experience a new creative visual. Like that, when they enter a hospital, they want to experience that health. Unfortunately, it is happening because many participants who are in the trial, they would like to feel a different when they want to participate in a trial. So all our strategies will be made. How does he enjoy this experience? And we are designing different things called as CLP, which was there in consumer product, consumer life experiences. We call it value experience. So many things are happening. On-demand health experience and rising expectations. Today you met a physician, you took a medicine, next day you don't expect the same response from him. You expect something different. You want, and they are more vocal now. So if the approach is in that way, that is what the healthcare is going to be. A lot of challenges. And the next generation is fully technology enabled. I think many of these things he has covered, so I would not like to do, but some of the practical things which we are trying to do is in the AI, all this, what we call it as a drug discovery, we evolve or we put our domain expertise into that, it throws out different options. It throws out different permutation and combination, and from that we pick it up. And from that is where the change in the industry professionals means we have to be geared up to do it. To give you a classic example for understanding, we do trials for maybe 1,000 patients or 2,000 patients, depending on that. But we launch a product. When we launch a product, more than crores of the population will be using it. So how do we know it? It works in everyone. We used to do what we call it as, that is what biostats is all about, parameters we used. Now options will be thrown to you. If you use it now in this shorter, this much population, on a wider population, this is the data. So that has come. And we need to be every day, I am saying it every minute, we need to be involved in this technology, involved enabled system. Please keep this in mind, it will all help you. Providing solutions to cater actual rather than perceived value. Perceived value is gone. I say, I make a product, I give a product, I perceive tomorrow it will be fine. I perceive. Come after 1 week, please bring a blood report. I will see the blood report. I will think that, okay, this will work like this, like this, like this. So the treatment or the traditional approach used to be on the perceived value. Now it's gone. We have to put across to the actual data. That's what whole approach will be. The patients will be empowered. And very importantly is that patient will choose the value-based delivery. If he is getting a value, he will take it. Just because my dad is using it, I cannot use. Just because my wife is using it, I cannot use. Means the awareness, I don't know, it has happened and we are facing this. Right? I can ask the new generation kids to come and lay down, I will put sphygmomanometer and I'll take BP. There you are, sir, put the watch, I'll get It will give time as well as what you want, they will say. Right? So that has become now, and we need to imbibe all those things. And industry is evolving through technology innovation, digital natives, and navigating these changes is a big challenge. Why? Once there is a big resistance within the industry, the talent pool. Because I read daily, I get adjusted, but not all the team will do that because they already spent 2.5 decades into that. They don't want to change it. So navigating these changes will become a biggest challenge where you all have a great opportunity to integrate it. So one, innovation is constant, so you need to upskill it for the growth strategies. Critical thinking, please critically think, don't just think. There is a big difference, right? When you're doing something, think. Everybody has got the same product, everybody, but only one product sells. Why? You have to critically think on that. The way that you, because I am bound by confidentiality, I cannot tell, but there are many innovative products which I am involved, and that comes from only critical thinking of the pharmacists like you, right? And then always don't be constant. Focus forward. You should be able to visualize. And the organizations which are able to link or think like that will be very successful and strategically grow. This is where we are having, I can say, 100% challenge is the talent. Though there are many people say there are no jobs, I'm not getting an entry, but from the industry, I'm again saying getting a correct talent is always an issue for us. Always an issue. And many organizers are spending close to 20% of their revenue in talent hunting. And that talent today has to be integration with science, technology, disease, everything. And analytical tools, we told many things, and actually practically we are using it. Without data, no decision is taken. Earlier we used to take decisions based on our experience, based on our gut feelings. If somebody might say, you walk from here to there, nothing is going to happen. But I have seen, what sir if I fall down? Do you have a data how many steps are there? Do you have a data how many calories I have burned? Do you have a data how much time I take? What if I am a hypertensive, if I fall down? So much of data today is being asked. And for that, analytical tools are important. That is what he was talking about, big data and all those. And very importantly, adaptability. You are not adaptable. If the industry is not adaptable, you stay there and strategies will not come. Because of the new diseases which are evolving, new dosage forms which are coming, all those things will be there. So hence I request, be a part of this sacred industry. It is in secret because we are dealing with the health. And for all these things will help in the strategies for the growth of the industry. We are all a part of that. With this, I conclude. With complex, try to make it simple. I'm not sure how much it has gone into your medulla oblongata. I'm not sure, but I've tried my best to put it very practically. All my 3 decades of experience, I've put it. And you know the industry, you know that industry is growing, you know industry is evolving, and what talent is required, what is there in the future. So plan your career. So anything, questions or anything is there with the limited time. I have taken 10 minutes extra. My apologies for that.
Yes. Volunteers?
Yeah,
Volunteers go with Mike.
Career opportunities, I will talk outside. If there is any career opportunities, I will take it outside. Because wherever I go, the first thing, sir, I don't have a job, do you have a job? Pardon, if it is apart from career, right? If you want to know what is happening and all, and more, please don't mistake me, because that's a common question whenever we go. Is pharmacy available there? Can I join there? So I know these are all the common.
I'm already working, sir.
Great. Sorry. You can tell your name. You can tell from where you are coming and ask. Just for my understanding.
My name is Ashwan. I'm working in biocon biology.
Great.
My question is, with increased number of biologics application and approvals from the FDA, what will be the future of generic? What about your opinion on this?
Yeah, great question. Please. For the other members, just to understand that if you go to my first slide, please go to my first slide if you can pick it up from there. Fourth one.
After during honoring, you put it.
Yeah. Don't enlarge it. Keep it there. Keep it. See, if you look at from there, in the segments we have generic as well as biosimilars and biologics. Biosimilars and biologics has a niche segments and the niche diseases which will improve the overall health or treat or cure. I can't say it is completely non-chemical based approach. There is a bit of chemical there because the proteins what they are using, there will be chemical based. The reason to choose this path, first and foremost, let me clear that both will be there. Generics will not go away. Generics will be there. Generics will not go away because there are many diseases you cannot address it through biosimilar path. The niche diseases which are in the oncology segment or in the autoimmune diseases will be more effective through biologics and biosimilars. Why? Because they use that path to make a product and deliver a product which will be lesser side effect, which will minimize the cost of treatment as of now, not all. Then and overall reduction in the life expectancy of because of the disease burden can be brought down. So those diseases are being targeted, as I said, market, product portfolio, wide segment, they think and strategize and go there. So chemical based or a generics will still be there. That is why even in biosimilars, we started using the word large molecules, complex biologics, simple biologics, right? Of course, there is a lot of innovation which is happening in MAP, monoclonal antibody release patterns, but cost is again a parameter there. Because if you have to take MAP, the cost of treatments will be very, very high. Of course, Biocon is in that direction to bring down the cost of treatment they are working. But it will take time for them to do that. Means many other companies, a common man can do that. But chemical based is one where the affordability is there. But with the chemical based, yes, there are side effects. There are some SAEs and AEs reported. Both will go. I'm not sure whether it will take 10 years, 15 years, or 20 years, but both will be there. And both have their own segments to address. Autoimmune diseases, as I said, oncology, yes, they are very, very, I can say, effective. But still, common man is not able to afford interleukins, erythropoietins, factor VIII. No, still, cost of therapy is where it is claimed. Both will stay. I hope I have.
Politics not to come forward so that they can see you.
Yeah, he is having one more question.
Stand here. There's a lot of back.
Hello, sir. I'm Dr. Siddharth, is a principal at the Siddharth College of Pharmacy, Bagalkot district. Sir, anti-cancer drugs are still very costly, not affordable to the common man. Whether industries are putting any strategies to get down the prices?
Yeah, I will not be able to tell industry is not putting it. Definitely industry is working, as I said, that oncology still is a point of discussion. We can sit down, yes, is a point of discussion. Why? Because the process involved in manufacturing of a product in oncology is expensive. You need to have many of the manufacturing criteria which makes it costlier. The API process is costly, and stringent clinical trial phases to be involved is expensive. So that's why the product is still not affordable to the common man. But government now what is telling is they are stepping up in taking some part of the burden financially. So if the burden comes down, then it comes under the DTCO, then the price. I'm not saying not all are affordable. Yes, there is affordability now to 40% of the medicine, which are being subsidized and coming under health insurance and all. Still 60% is out of that. Common man still is having a problem in getting an affordability for that. So from that perspective, industry is working in a very, very big way in terms of dosage, so that the cost can be reduced. The cost usually comes in the number of cycles. I am not sure whether you are following the oncology therapy. The methotrexate or melphalan therapy, when it is done, they used to run 16 to 17 cycles. Now it has come to 6 cycles. So that when the chemotherapy cycles come down, the cost will be coming down. But still, not to the level where it is a common medicine like this. There are some diseases which are still requires that kind of thing. That is where biosimilars and biologics will be entering. So the more it comes, then affordability will increase. The maps are going to come. So yes, instead of paying 100 rupees, it may come to 60 rupees, but still 60 rupees is there. 40 rupees has come down.
Of course, people are getting from Pradhan Mantri Jan Aushadhi the reduced prices.
Yeah.
So through this platform, I request central government, even every medicine should available, including anti-cancer drugs, anti-TB. Every medicine should be included.
That's the dream.
Yes, sir.
That's the wish. And I think most of the pharmacists who come here to do that work.
Thank you, sir.
Maybe we can allow last one question.
Hello, sir. I'm Dr. Kavita, professor with the College of Pharmacy, Bangalore. I want to talk about this artificial intelligence. I was very happy about that innovation. My target is what is my doubt. Maybe the innovation should be the cost effective.
Innovation should be
Cost effective. Whether this artificial intelligence will be a cost effective.
Next speaker
I don't
address
know,
your
we are replacing the manpower
He's not
because
belongs
of
to
this
this
artificial
organization.
intelligence. What about the job opportunities of the already existing persons?
Go to the
It may affect. Sorry.
welcome
I'll
slide.
tell you that please take away that here artificial intelligence is going to replace man. No. Job opportunities. That's why I say that you have the talent to integrate science with the technology, with the advanced process. Hello. Yeah. So if the integration is there with the talent, we will always have it. Artificial intelligence is the data which is being accumulated and it will throw you off. But somebody has to decide. Right? Somebody has to feed it. Somebody has to evaluate. Somebody has to analyze. Somebody has to do action. So the jobs will always be there. But there is an integration. Only the correct talent pool will stay there. You got my point?
Yes.
Right? So this is what the development is all about. Development will always happen at the cost of something, but doesn't mean that it will completely replace. It will not. Maybe 10 steps we were taking earlier, now it has come to 2 steps. But somebody, domain expertise will always be there. If that would have been the case, all pharma would have been closed and only IT sector would have been moving
Yes,
around
that was
everywhere.
my-
It will not happen like that. Even if you look at any IT sector, the domain expertise team who are there in data management as well as in many other manufacturing sectors are the core people who have been on the floor and worked and come there. Because you need a structure to be, and that structure comes from us.
Yes, thank you so much.
Right.
Yes.
Don't have that fear. That's what in the first thing when I saw Dr. Rajesh slides, please don't get fear that should I join IT? Why did I come to pharmacy? No, it will not happen like that. But it is not something you can prevent. This has come. You improve yourself. How do you do it? You have the domain knowledge. Nobody can take it away. And all these softwares are going to come to enable you to give a better solution. It cannot replace anything. It cannot. And for that, you have to be up for that.
It should not happen, sir.
Yeah, yes, there is a hype, there is a use. Today that ChatGPT is making hell of a noise. Yeah, it will all be there. We have to live with it, and we have to get ourselves gate for it, and still prove that human interference is still there.
Yes, sir.
There are no questions. Thank you. You have something?
Yeah, please.
Yeah, please. Sorry.
Sir, this is Dr. Fuller. So my question is a bit related to pharmaceutical industry, but not exactly related to the talk. This is related to the consumer. As we see most of the time in the market, there are some few combinations are coming. This drug, that drug, combined, that drug. The rationality of these drugs, doctors sometimes refuses, but pharmaceutical industries are promoted. So what is the actual standard for rationalizing these medicines? How they are making it?
This is a regulatory question. Always the physicians and the manufacturers will have this conflict. And always the drug combinations which are there are more in India, and sometimes it is crude. That is why the added benefits or the synergetic effect of these combinations are more. Why? Because so that the dose and the treatment comes down. If I use a single, the therapeutic window is larger, and if you use combination, I can still give the same therapeutic window but at a less duration. With that concept, it comes. But the rationale is nothing but to give a synergetic effect or a synergetic pharmacological effect. Now, the regulators now are very strict, which is rational, which is irrational. Even though the products which are available, if it is irrational but still being in the market, now they are being banned. Stop. Ultimately, the regulator will look whether this is irrational in terms of the end user because it is causing serious adverse event or SAE on a long-term use, or there is an high incompatibility because of non-stability of the product. Products have been recalled. You can see it in the market. You can go into a drug information center. You will see how many combination drugs have been recalled. But yes, you will see some irrational combinations. Right? We saw diclofenac with paracetamol with mucilage. So many things will be there. But yes, now regulators have become more vigilant, and such kind of irrational drugs are being recalled out after being in the market for many years. But that is why I am saying it is the responsibility of all you people to become a vigilant pharmacovigilance guy? How many of you see the label or a pack insert and read and do it? Do you do? You don't do. Please do it. Whenever you buy a strip or a product, see the content is as per the label, see the color of the tablet is as per the label, and if it is not, or if there is no tablet, after you consume, you experience some kind of an AE, there will be a toll-free number there. How many of you call? Nobody call. Please do it. Everywhere it is there, very strong. Today I am doing pharmacovigilance business, which is giving me close to 80 to 100 crores. Only by doing this. Everywhere they are vigilant, only here. But now please start doing it. And government is also encouraging it. Government is having PVPI portal, which was not there earlier. Not now, everywhere it is there. And pharmacovigilance system has to be there in any product launches. They have to do. And when you do, more such events keep coming. That's where the regulators will become vigilant from this portal. They will take it for analysis. They will consult the subject expert people. Then they will recall it. But if we don't do the pharmacovigilance part of it, or we don't complain about it, it will be there. Right? How many of you do the protocols and all? You are all there in the protocol. It will be there. Adverse event, serious adverse event, contraindications, all those things will be there. But how do we follow it in when the product is being consumed? If something is happening, you should report. Please be vigilant on that. Henceforth, please do it. Whether the packing was open, whether the packing sealed is not there, whether the batch numbers are not there, manufacturing date is not there, expiry date is not there, color of the tablet many a time it will be different. Then the toll-free number, please register a complaint. The more we do, it becomes an action for the regulator. Is it fine? Joshi has got one more.
Sir, the session was. Yes, sir.
No, he wants to say something.
Sir, the presentation and the session was so wonderful, and I have one doubt, so which might be maybe different from this scenario itself. So, the strategies that you have told for the evaluation of the drug and the treatment of the diseases. So, if the strategies and everything is working in the same manner, so if power and money, if brain and money comes together, so are we not creating the diseases and we are selling the product?
Not on this podium, I'll give this answer. I'm standing here as a recognized society because of the industries and strategies. If strategies are not there, then the chief strategy officer is not required at all.
That's why I told you, sir. Thank
Right?
you, sir. Thank you.
Diseases will always be there, but will not create any disease. Yeah, I understood. Because see, print media and other things are always there, accessibility is there, there are things, there are situations. If not something is not there, means why will we ask this question? Yes, one-to-one I can tell. Because they are videographing me, they are listening to me. And if you give permission, yes. See, I don't know, yesterday you would have listened to him, Padkar sir is there. I don't know whether he recollects me. I worked with him in the starting of 1990s. He was the first person to bring the bioavailability and bioequivalence studies to India. He was the first person to tell to our regulators how the protocol of BAB studies should be. You have such a pleasure of being such a personality. Today, all generics are doing, and all clinical research organizations like us have done studies. It is only it is guidance how to write a protocol, what are the contents of the protocol, what are the regulatory requirements, and I was the first student of it.
Thank you, sir, for gracing our stage with your mind-blowing ideas.
I request Dr. Bharati and Dr. Prakash Vodandanavar to honor Mr. Raja Agnihotri.
Now it's time to take break. So I request all the dignitaries and delegates to proceed towards the college quadrangle. Let's resume in 10 minutes. So please be back by 12. Thank you.
Yes, sir. Yes, sir. ಸರ್ ಡೌನ್ ಆಗಿರ್ಬೇಕು ನೋಡಬೇಕು ಯಾರ್ದದು
ಇಲ್ಲ ಹೊಸ
ಸಿಗ್ನಲ್ ಎಳಿತಲ್ಲ ಮತ್ತ
ಹೊಸ
ಇಲ್ಲ ಅಪ್ಡೇಟ್ ಮಾಡಿದಾಯ್ತು ಸಿಗ್ನಲ್ ಎಳಿತದೆ ಫುಲ್ ಇದೆ ಆ ಸಿಗ್ನಲ್ ಯಾಕೆ ಇದಾಗ್ತದೆ
ஸ்லைட் சேஞ்ச் அப்படி வாங்கிட்டாங்களா?
எஸ் ஓகே
தேங்க்ஸ் சார்.
ಇದು ಇಲ್ಲಿ
ಏನು
நான் கால் பண்ணவே இல்லை. டே 3 வேற போறாங்க அந்த கான்ஃபரன்ஸ்லயே. டே 3 வித் டாக்டர் கீதா அண்ட் பாரதி. இப்போ நான் எப்படி கால் பண்றேன்? மண்டே காலையில பேசணும். இதெல்லாம் கன்ஃபார்ம் ஆகி மண்டே இது பண்ணலான்ட்டு தான் அவங்க கேட்கணும். பண்றீங்களா? பண்ணலாம். எல்லா இன்ஃபர்மேஷனும் பண்ணி ரெஸ்பான்ஸ் பண்ணி வச்சாங்க. இன்ஸ்டாகிராம்ல வருது. காலையில அந்த போஸ்டர் அவங்க இன்ட்ரெஸ்டட் தான் இருக்கேன்னு சொன்னாங்க.
Session start.
Welcome back. For the third keynote session, we have an international speaker, Dr. Devendra Ridurkar, subject matter expert, complex formulations, Urax Pharmaceuticals, Barcelona, Spain.
Sir, he's here to brief on fundamentals of 3D printing technology and its applications for patient-centric therapies. Over to you, sir. Hello. Sir, you can proceed.
Okay, can you hear me?
Yeah. Yes, sir.
Okay. So good afternoon, everyone. First of all, thank you very much for inviting me for this conference. And so today we'll discuss about the fundamentals of 3D printing and what are the applications specifically for the patient-centric CNS therapy. So in this, we'll understand. So can you see my slides?
Yes, sir.
And I'm audible too?
Fine, sir.
Okay, so during this half an hour sessions, I would like to go through what are the automations that we are using in the pharmaceutical industry. What you mean the treatment personalization? It's better to understand what is treatment personalization. What are the different options or product design for patient-centric dosage form? Then I will give the brief introduction of 3D process, their technology, their applications, and what are the companies they are working on 3DP and the products which are available in the market. So first, I would like to start about the automation in pharmaceuticals. So at present, we are using the automations in order to develop the robust formulation and the patient-centric treatment and new discoveries. For example, to treat the end users, for example, patients or caregiver or healthcare provider. Also, we consider the geriatric and the pediatric peoples, and also in order to get the patient adherence and the drug safety. In addition to that, the automation is generally used for batch manufacturing to continuous manufacturing. We are nowadays, we are using the robots, and also we are using the process analytical tools. So let's understand what is patient-centric treatment dose personalization means. As you know, the dose is changed as per the requirement. Like for the kids, maybe it is 10 milligram of the same drug, but for the adult, it may be 100 milligram. So it changes as per the requirement. For geriatric people, it is different, and for the adults and for the kids, it's different. So that's the reason most of the companies are now, they are working on the dose personalization. Dose personalization means as per the requirement of the patient. So if he need 1 milligram, we can prepare the tablet of 10 milligram. If the patient need 100 milligram, so he can print at his home the 1000 milligram of tablets. So I will explain, I will show some video graphs, so you will understand much better. So this is a patient. What we do, so like in dose personalization, we give the accurate dosing by using some multiple drug fillings. And because of that, we can improve the patient acceptability as well as the better pediatric acceptance. We can control drug release by using the single drug, or sometimes we can use the multiple pills. As this can be printed at home or even at the pharmacy or even in the hospital. So you don't need to prove that our formulation is stable for 3 years or 4 years, like for example, for that we used to do for the conventional medicine. But in case of 3D printed drug products, it will be like a week is enough if it is stable for 1 week, because you can print when you want to take. And there are also some personalized medical devices and the drug eluting devices in the market. And also we can use this technology for personalized DNA and RNA-based therapy. So now let us understand what are the drug product designs that are generally formulation scientists use. First one is the fixed dose combinations. So you are, maybe most of them you are aware that there are, like for example, paracetamol gives along with some other NSAIDs or some other drugs. So like 2 or 3 drugs are mixed together and they are known as a fixed dose combination. But generally it is used for the anti-diabetic, cardiovascular systems, or for respiratory or HIV. But as we are using the multiple drugs, the dose is too high. And as this patient-centric drug design are more specifically useful for pediatric as well as for the geriatric persons, and swallowing is very difficult for such kind of people. So this is not recommended for the patient-centric drug design because of its size. Then the second option is oral dispersible tablets that you can dissolve in the water and you can drink it. And especially it is for the CNS drug. But the disadvantage of this technology is the production cost is very high, and as it is used with the drug is having its code beta test, so it's very difficult for the kids or for the geriatric users to accept the test sensation. The third one is the multi-particle formation, that we can use the mini tablets or the pellets, but the technology is too critical and it's very difficult to handle the weight control, continuum property, and also the patient handle is different. So that's the reason, considering this disadvantage, the pharmaceutical technology is developed and now they are using the 3D printing technology. So the advantage of this technology is it is versatile and flexible, and you can give a dosing to a single patient as well as multiple patients as per his or her requirement. Manufacturing possibility, we can manufacture at industrial scale level, you can manufacture at a pharmacy, as well as you can manufacture at patient hands. You need just the printer and the computer. What are the advantages for this 3D printing, particularly for the patient's perspective? So the persons who are having the swallowing difficulties, for example, the geriatric people or the children, this technology is very useful for them. And considering the business, like for the pharma industry, that we can build intellectual property, we can build patent technologies, we can differentiate, we can develop the differentiated products, and also if there is some patent hurdles in order to make generic formulations, we can use this technology. Ideally, the 3D technology was invented in 1984, but in pharmaceutical, the first product was approved in 2015, that is preterm. And there are different processes that we can use for the 3D technology, that stereolithography, selective laser scintigraphy, or physio deposition modeling. So I will explain in the next slides about all this. So why 3D is gaining more attention in pharmaceuticals? Because there are lots of challenges in the conventional manufacturing process. What are the challenges? For example, when we start the conventional process, we need to mix, granulation, drying, milling, lubrication, and compression, and it takes a long time. Number of people are involved, and because of that, the quality of the final product or the drug stability is not so good, and also the long processing time, and also it's very expensive and needs lots of equipments and the manpower. And additionally, there is a 20 to 30% of material is wasted. So that's the reason 3D printing has been initiated, and now at current, the global 3D printing market is around $278 million, and we are expecting that it will grow to $522 million by 2030, and the growth rate is 6.5%. The majority of this work is going in the US market, and the Europe is on the 4th level. Even India also, now most of the companies, they have started working on 3D printing technology. So now coming on the advantages of this this is a single solution for various problems. As a formation scientist, I face day-to-day various problems. The first problem is there is a lots of API, particularly in NCEs. When we start working on NCEs, around 60 to 70% molecules are water insoluble. They are either class BCS2 or class BCS4. And because of that, we always get the viability issues. So 3DP, we can solve. So we can enhance the solubility. Even we can generate the rapid disintegrating tablets, and we can enhance the dissolution. And as we know, if we increase the solubility, there is an enhancement of the viability. Second problem that a formation scientists face, that stability during the API processing, that there is API which are unstable during the granulation, or they are aqueous problem, like they are hydrolytic states, or they don't need the heat. So this is the good process in order to avoid the API stability. The second is patient-centric requirement. So we can prepare the drug product according to personalized regimen as per the requirement of the patients. If there is a high dose medications, we can prepare the rapidly disintegrating tablets, and which is very easy to administer with the liquid. If the taste of the API is very bitter, there are several drugs which have the bitter taste, or the taste is not good. So we can convert it to the test masking formulation by using 3DP technology. So this is the general process that 3DP works. First, we need to model. We need to model into the computer or the laptop by using the CAD, computer-aided design, or any animation model software. Then you give the command for the printer, and the machine will read, and she will start laying down the successive layers of liquid or the powder to build the tablets or any other solid dosage form. And then we need to finish, finishing like the make it like a very decorative. So there are 4 processes in order to develop the formulation by using 3D printing. The first one is the stereolithography. In this, it's like creating 3-dimensional objective, and in this we use a laser to build the required structure. Required structure means like a tablets, and we use the layer by layer. So I think I will show you some video so we'll understand it. So now the drug layering along with the polymer was tried with the laser. Now we can see the tablets manufactured by the stereolithography by using the laser. The next process is inkjet 3D printing. In this, we use the different combination of active ingredients and excipients, particularly the inks, and then they are prepared, sprayed onto the small droplets.
Making healthcare personal.
This is like spraying with the liquid, and you can see this. The third process is the selective laser sintering. This is a blend of drug along with some excipients. So, as I told you about these three processes, they are not that much used in the pharmaceutical industry. The most used method is the fused deposition modeling. In this, we use the hot melt extruder connected with the printer. And what happened in this method, we use the heat in order to melt the polymer, and then it is extruded through a movable heated zone. And then we are formation there, we form layer by layer for in the angle of X by Z stage. And generally, we use the CAD in order to develop this formulation. Generally, this technology is useful for the amorphous formulations or drugs that have poor solubility. Also, we can use it for the implants. This technology is very good for the oral complex immediate release as well as the modified release formulations, polypills, or drug-loaded microneedles. I'll explain it in the presentations via video. So I'm preparing the 3D printing of the thiophyllene tablet. You can see here, this is a nozzle head. The polymer along with the drug is already started melting. Now it will start printing of the tablet, and it's too fast. So because it's as per the requirement, you can print. Now the liquid is solidified, and here you can see the tablet. Now coming on what are the materials that we generally use and what are their characteristics? So the first one is the powder characteristic. In order to have a good flow, the particle size, shape, porosity, and crystallinity are very important. Water content should be in optimum level. It should not be high because it will affect the flowability. The density and the flow and uniformity of mixed material should be fair enough, or it's be good. In case if you are using the fluid, the print fluid may be like a liquids or suspensions or hot melts. It should have good rheological properties or viscoelastic properties, and it should be stable in the high heat. Always there is a risk for every manufacturing processing. So 3D is not an exceptional case. So there are various manufacturing risks. For example, we can get the variable layer thickness, but always there is a control strategy. So if there is, you found that there is a variable layer thickness, you can always control with the software. Sometimes there is an inaccurate position during the printing or printhead clogging. So you need to work out on the raw material or the real-time layer thickness monitoring by using the process analytical technology, that is online analysis. Sometimes there is a friable tablet, so you can change the powder deposition rate or roll speed. Most of the companies, they are working to develop the polymers. For example, Merck, they have various polymers, BASF or Unique, they are working on the development of the polymers for 3DP. So at present, the polymers are cellulose, like for example, hydroxypropyl cellulose, HPMC or Udazit L100. We can use this for generation of these 3DP processes. And I have used several drugs as a model drug by using these excipients and the drugs, like for example, acetaminophen or indomethacin, theophylline. These are the polymers generally useful for the 3DP technology: polyethylene glycol, hydroxypropyl cellulose, HPMC, polyvinyl pyridone, and the copolymer of ethylene oxide and propylene oxide, for example, Lutral F127 or Lutral F68. The most useful polymer is Collidone V64 and Soluplus and Lutral F127 because of its flexibility and the processing time and the processing temperature. In order to reduce the processing temperature, I always use the plasticizer, which will give the more flexibility as well as the plasticity, and it will reduce the processing temperature. There are different types of plasticizer, for example, citrate, fatty acid esters, like triethyl citrate or butyl stearate, or vitamin E TPGS. That is also one of the excipient of choice. Now coming on the disadvantage and the advantage. So if we see the advantage, we can give the high production rates due to its fast operating system. High drug loading is possible. We can reduce the material wastage, and also this is a good cost-saving options in the production. It is very good technology for the water-soluble drugs. It saves time and cost. And the most important thing is the customization as per the patient requirement is possible. And the low production volumes are feasible and enabling its personalization. Coming on to disadvantages, the machinery cost at present is too high, but now compared to 5 years, now at present the cost is coming down, and maybe in coming years it will go further. It will go down further. It's not suitable for if you want to develop 100,000 tablets, so it's not suitable technology for this. It's difficult to remove or install manually. There are some mechanical issues, and the production of dangerous atoms is not possible. These are some examples that 3D printing is used in the pharmaceutical drug delivery systems. So as I told, the fluorescein was developed by using the hot melt extrusion technology, aspirin, ethanol, and ramipril. Go next. In the earliest slides, I told that there is a product which was already developed, and it is known as the sputum, and it was developed by using the 3D printing technology. It is available across the globe. It is oral dispersible tablets for levetiracetam, 500 and 1 gram. So the Aprecia Pharmaceuticals has developed this first 3D drug product based on the ZipDose platform. In ZipDose, it enables the high-dose medications for a rapid disintegration from, and it uses the product is layer by layer without using the component forces, punches, and the dies. So So the key players in 3D printing, mostly that's, I told you, Aprecia, they already have the products, and now they are working on around 10 products, and in coming years, they will come in the market. Additionally, GSK, J&J, Novartis, Pfizer, Teva, Durax, and these are the companies they are working in pharmaceuticals. And for machineries, the HP or 3D printer drug machines, Fabrics and Catalan and 3M drug delivery system, they are working on the machine parts. So I hope you understood the basic of these 3D technologies. And thank you very much for listening to me. And if you have any questions, so please.
Thank you, sir, for inspiring these young minds.
The speaker of the keynote session 4 is Dr. Karthik Anantaraman, Vice President, International Apollo Hospitals.
Sir is here to conduct a session on practical use cases and digital transformation in pharmaceuticals and hospitals. Over to you, sir.
Am I audible now?
Yes.
Yeah, sorry, it took a while. I couldn't unmute myself. I think I needed help from the host to unmute me. Once again, good afternoon to all of you, and thank you so much for inviting me to share a few of the experiences that we've had in terms of practical implications of, technology in health. I would like to focus on practical applications that we have seen in our experience in pharma, in medtech, and in hospital services delivery. Which is actually, and tech, as you know, is one of the most sought after topic of discussion by patients, by physicians, alike and by health tech entrepreneurs and enthusiasts per se. What I've tried to put together is a small presentation that actually outlines the practical use cases and applications in healthcare. As I mentioned to you, from a pharma setting, from a setting in terms of medtech and hospitals. So I'd like to walk you through this presentation and more than happy to answer any questions at the end of it. So once again, thanks for this opportunity. Please let me know, organizers, if my full screen and slide is visible and I can start my presentation.
Yes, sir.
Thank you. Wonderful. So having said that, so naturally, if we were to look at the way how digital health has been evolving, so broadly, I'd cover this topic under two headings. One is digital health trend and leveraging digital health initiatives. Just give me one moment, please. There is a little bit of a noise. I'll try to take care of that. Am I audible now? Can you hear me?
Yes, sir.
Yeah, I'm sorry. I had to step out. A little bit of a drilling in the neighborhood, so sorry for that. So yeah, broadly, I will focus on digital health trends and leveraging digital initiatives to drive the market growth. So these are the two main topics that I shall focus on. Broadly, if we were to start with a quick overview of the market, as the previous speaker was also mentioning, there are many accomplished organizations who are in the field of medtech who have actually transformed themselves from a hardware organization into what we typically call as a technology transformed or a connected technology transformed organization. And you can see all these names given over here. And as you can see, the market of connected health is actually growing at a significant pace of almost 21.9% CAGR. And by 2027, you will see that this is expected to grow to a total of $109 billion in terms of overall market size. Within this, if you see between wearables and non-wearables, traditionally you would see that there was a trend of wearables which kind of got depleted over a period of time. But now again, post-pandemic, we are seeing that there is a significant increase in connected tech, where not only is it important to monitor health of patients when they are in the hospital, in the care of their under the care of their doctor and treating nurse, but it is also important that a lot of hospitals have now started to focus on what we call patient-reported outcomes. Patient-reported outcomes is a methodology where we are actually providing technology in the hands of a patient to report new efficacy-related signals, new safety-related signals when the patient has left the healthcare system and gone back home. So therefore, it's a new way of looking at clinical outcomes, measuring clinical outcomes in the practical healthcare setting. And that's enabled through tech. And that's where we see a significant growth that's happening in the wearable tech market. At the same time, you can see the adoption of connected health overall, be it connected health at an overall level, aggregated level, or wearables and smartwatch type of tech is significantly growing. And we're seeing a huge difference, pandemic playing a lot of huge role there. Just to give you a brief insight there, a lot of companies starting with the likes of Practo, Teladoc, etc., who were actually known for their consultations, digital consultations, etc., they saw almost an 8 to 10-fold increase or growth in their teleconsultation traffic just with the advent of the pandemic. And that has sustained over a period of time. The new patient convenience and the new doctor convenience that we have seen post-pandemic is earlier, even for a routine health checkup, or if it's a diabetic or a patient with high blood pressure, even for a routine checkup of blood pressure and sugar, they used to go see a doctor. Now, both doctors and patients are completely in sync that only if it's a first-time diagnosis which requires a one-on-one personal intervention I'd like to go see a doctor. But otherwise, if it's a routine consultation or a checkup, I'd like to do it over the net. I'd like to do a teleconsultation or video consultation. We see that complete change in consumer psyche, consumer preference, which is a growing trend, and that will continue to stay. Therefore, we are seeing that across medtech as devices, supporting health tech system, which is typically what we call healthcare IT, which is software and system, and across services, there is a significant growth that is being seen. Where is the entire digital tech in pharma and medtech heading towards? Broadly, earlier, if you were to talk about digital transformation or health tech or digital health as we call, they were classified under 4 pillars. One is monitoring of disease. Second is diagnosis of disease. Third is all technologies and tools that will help in treatment decision-making for diseases. And fourth one is continuity of care, which is for ongoing care. But the one area which I have highlighted here on the slide draws special attention, because digital therapeutics is one of the most recent advances that has happened in health tech technology. Consider digital therapeutic as a digital health intervention for a patient who has been prescribed a pharmaceutical drug. Now, by adding digital therapeutic along to the doctor's prescription, you are able to significantly demonstrate that there is incremental clinical outcome. What I mean by that, and I'll prove it to you with an example. I'll show you one of the recent published evidences on this from none other than Dr. Shashank Joshi, the chief of endocrinology, one of the senior most endocrinologists in the country, practicing at Lilavati Hospital, Bombay, where he's clearly showed that when he prescribed digital therapeutic solution to his diabetic patient, in addition to medicine, when the patient adopted digital therapeutic in the way he managed his diabetes, he could see that the patient's HbA1c, as we call it as, which is basically the amount of sugar that is basically attached to the hemoglobin in the blood for over a 3-month period, the amount of HbA1c and also the fasting blood postprandial sugar, it had an incremental reduction because digital therapeutic was used in the overall way of treating it. Therefore, very clearly we are seeing the role of the advent of digital therapeutics changing the way how chronic diseases are being managed. So that, again, thanks to technology. So therefore, there are multiple digital health tools that have been developed, in this direction. You can see starting with digital therapeutics to consumer wearables, connected biometric sensors, smartphone cameras, clinical trial related tech tools, in-home connected virtual assistants, telemedicine virtual visits, personal health record, web-based interactive program, health disease management app, and consumer mobile app. This is the entire universe of all technology-related interventions that one can have, keeping patient in the center. And pharmaceutical companies have a role to play in almost across, the entire spectrum of this. All of this is primarily driven through what we call healthcare consumerism, where patients are no longer looking at healthcare from the angle of just going when they have an issue. A lot of people are now taking proactive measures to take care of their health. And now they are extremely quality conscious. Like how one would go to an Amazon and basically say that this is the quality of service on a 5-point rater scale or a 5-star scale. Now they're able to go and rate the quality of services they got from a doctor or from a hospital. And that's the recent change that we have seen in terms of looking at healthcare from a B2C or a consumer angle. Now, what are the drivers of this growth? Naturally, this tech growth, this growth in healthcare consumerism is primarily led by chronic diseases. And in chronic diseases, cancer, diabetes, high blood pressure, or heart disease, as we call, are some of the leading drivers of this trend. This is a typical Gartner's chart, as we call, or the hype cycle, as we call, where it clearly mentions what are those health technologies or digital health technologies which are in a mature stage and which will also stay put for a longer future, and what are the ones which will possibly slowly die a natural death. So you can very clearly say significant progress expected. If you see, that starts with 3D bioprinting. It looks about biomarkers. It talks about artificial intelligence in drug discovery and drug design, which is already a reality. It talks about at-home lab investigations. It talks about 3D printing of drugs, medicines, capsules, tablets. It talks about artificial intelligence in diagnostics. It talks about portable diagnostic devices. Remote robotic surgery is already a reality across multiple applications. Now, augmented reality in surgery and chatbot in health applications will be the next one. Employee wellness programs, and digital health insurance. These are the various technology solutions that have been projected to be the ones that will make significant progress in the near future, something to watch out for. And it's very clearly documented that consumers and patients are very much looking forward to adopting technology in the way how they wish to receive and consume health service. And that's been documented from both rural and urban areas. So quickly, I will go into how the government has responded to this. So in our country, a lot of the tech is regulated by the CDSCO, and the pharma is regulated by the DCGI, which is Drug Controller General of India, under which they have now created 4 major verticals to ensure that health tech is reviewed and from a regulator perspective approved in a very structured manner. And this basically outlines the structuring that the way how government has also structured itself to make sure that there is a faster adoption of health tech in the country. Having said that, what are the newer technologies that are disrupting the way how we deliver healthcare? Under digital health, you can actually see the more evolved ones are in cardiology, critical care, women and child health, home care, imaging, and lab technologies. I'll give you examples of that. These are all technologies in cardiac. You can remotely monitor an ECG, the heart rate, the heart rhythm. Someone is having a heart attack. You can actually look at, using this on an aeroplane, on a train, on a bus, any mobile platform, immediately picks up a signal if the patient is having breathlessness, chest pain, anything. Immediately, it will tell you if it's an emergency. You can rush to the nearest hospital, save a life. If it's an ambulance, it can do a remote consultation connected to the internet with a video consultation. For patients with heart failure, this is a life jacket or a vest kind of a wearable which measures cardiac output. Sensors, there are sensors which remotely measure cardiac output. It can send an alarm signal if the cardiac output is falling, so the patient can immediately be revived. So all life-saving tech-related solutions that are there. Similarly, you have solutions developed by scientists in Singapore called the Apollo program, looking at AI-driven a national platform for CT coronary angiography for clinical and industrial applications, very clearly establishes the extent of blockages that are there in the coronaries, which can help save a life through early detection. And then ultrasound can be huge. It has an application if someone is having a bleeding in the abdomen, it can immediately detect. You can do a live remote telecast of the image, seek a second opinion from a specialist radiologist, get an immediate opinion, and quickly save the life of a patient. It has applications. It has applications in chronic care to measure blood glucose, to measure blood pressure, to measure activity, and seek help from a chronic disease specialist. It has applications in critical care, where if there is a patient admitted in the ICU, you can live relay the critical care parameters from the ICU monitor to a critical care specialist anywhere in the world, get an opinion, and save a life of the patient in the ICU. It has applications in home care and women's health, where women after giving birth, they have a laxity of the pelvic floor. It can lead to urinary incontinence, and you can measure the urinary incontinence, manage it medically, and you're able to provide a normal life in the postpartum phase. And this is a very interesting application in neuroscience, where technology can help manage mood as well. It's a wearable patch that you wear on the forehead. It helps modulate neurotransmitters in the brain, and this can actually have a very nice application in terms of managing patients with neurological imbalances or neurological disease as well. And last but not the least, multiple technology interventions available in the laboratory healthcare system, where you have connected labs today, where you don't need a specialized pathologist or a senior lab technician to be running a lab. You can have people with basic lab technician qualifications run a lab, connect the devices, get the parameters from the device, connect to a central command center where you have experienced pathologists and operational scientists who can then read and report out, and hence you can run a digital lab network using that. And all of that is enabled today. Last but not the least, this is the commonest thing you will find at home. There may be relatives, parents, friends who may be diabetic. They'll be using insulin. They'll be injecting insulin through an insulin pen or an injector that looks like this, or through a syringe. Now you have Bluetooth-enabled insulin syringes that will tell you exactly the amount of dose you have to take so you don't go into low blood sugar. It will help you monitor your blood sugar through a connected glucometer, so you can monitor your blood sugars very well at home as well. fantastic new startups based in India. Some of these are based in Bangalore and multiple other places. SIGTuple is an artificial intelligence enabled pathology lab. You can set up a very nice remote pathology lab anywhere in the world. It will read, it will compare the images using an artificial intelligence algorithm. It will give you a provisional diagnosis, which is finally endorsed by a clinical pathologist. HealthCube, another critically acclaimed funded health tech startup, Bangalore-based. They have a full-fledged 25 diagnostic tests to be run from a single portable system that you see here, connected to a tab, and you can run it like a remote pathology center with reporting services as well. Finally, digital therapeutics I mentioned to you have applications across. You can have every organ system, every type of chronic disease is covered here, as you can see. There's a lot of research and a lot of applications going there. I mentioned to you about a doctor who used it very well. These are the multiple established companies that are there in digital therapeutics today, and each one is focusing on a specific organ system or a specific type of disease or disorder that you can see, very well established and doing excellent work there. This was a publication from May last year, where Dr. Shashank Joshi, I mentioned, one of the senior endocrinologists of the country, presented a paper at the American Association of Clinical Endocrinology in the US in San Diego, where he showed that how a digital therapeutic, a digital technology platform, which was developed by a startup based in IIT Madras, helped patients achieve remission of type 2 diabetes. So therefore, digital therapeutics are really forming an integral part of chronic disease management. It is here to stay across not just diabetes, but multiple therapeutic segments and areas that I mentioned to you. And the last part of the presentation I want to show is that digital biomarkers is a reality. It is not only finding applications in research, it is also finding applications in drug disease delivery and disease management as well. So therefore, that is going to stay. It is starting with Alzheimer's disease and some of the neurological, neurodegenerative conditions is where some of these applications are finding a role. And finally, how to make this sustainable. I think this whole digital health as a concept is sustainable if we can work together. Now, who should work together? Pharmaceutical companies, digital health technology companies, hospitals, and treating doctors or physicians come together as an ecosystem to make it work. It does need support from pharmaceutical organizations, which I'm going to show you here, where each company already has a partnership with some of the established pharma companies here, like Boehringer, Novartis, Pfizer, Roche, Merck, across various therapeutic areas. And this kind of a partnership is the way to go, where a pharma and a tech company come together in the betterment of healthcare and to deliver the technology to patients who need them. It is partially funded by pharma company, partially funded by insurance, and that is what makes some of these very sustainable. So I thank you for your attention. My coordinates are here, my mail ID and my mobile number is here. This is the end of my presentation. I hope I was able to share some insights about new trends and technologies from a practical application and a use case perspective that is currently happening across pharma, medtech, and hospitals. Thank you for your attention again.
Thank you, sir.
We are really lucky to have heard from you, sir.
My pleasure. Thank you. I'm more than happy to share this presentation. If the presentation is of interest to all the delegates, all the colleagues on this call, I can share it, and please feel free to connect. But thanks for the opportunity.
So just 1 minute, one question is there.
Hello, sir. It's Dr. Kavita, Professor, Pharmaceutical Sciences, Bangalore.
Good afternoon.
Very happy, sir. Thank you so much for giving the latest information. Thank you so much for giving this valuable information to the recent technologies. And you spoke about that insulin, sir, regarding diabetes. So in the diabetes, the thing it is the injections. Still patients are facing the problems. with that high risk of taking the medicines by injections. No oral therapy is not available because a lot of publications I'm seeing regarding that work on oral insulins. So any new technologies when it will be exist into the market, this oral insulin, sir? Is there any idea? Hello, sir. Am I audible?
Sorry. Am I audible? Can you hear me?
Yes, sir.
Okay, I think I had gone on mute. Sorry. So yes, that's a very good question. Yes, there is a research that is being currently pursued by Biocon. Okay,
Yes.
and all of you may know Biocon is one of the leading biotechnology firms from India. It's one of the largest biotech firms in Asia. I had the privilege of being a part of the Biocon family some years back. So they are researching on what we call oral insulin. So it is a new technology where if that technology were to be researched completely, and if it were to be successful in completing phase 3 clinical trials, insulin will now come in an oral tablet form after that, subsequent to that. And then the patients will have a choice of either injecting the insulin or taking insulin in a oral tablet format. Currently, this product is undergoing clinical trials. It will still need at least 3 to 5 years of research before we know whether it is going to be successful or not. But it is a research that is progressing in the right direction.
Okay, sir. Thank you for the information, sir. And one more small clarification, sir. Regarding the digital therapy, discussed a lot. So I want to know about that fulfillment requirement of, it is fulfilling the requirement of the patient compliance?
Yes, yes. So the digital therapeutic addresses multiple things. One, beyond the medicine, as we call beyond the pill, as they generally call, it will address the need for dietetics, nutrition, physical activity, compliance to therapy, and compliance to repeat investigations, which will tell us whether the medicine is working or not in the dose being prescribed. So it is a holistic approach to ensure that both pharmaceutical and non-pharmaceutical supportive approach put together gives a collective incremental benefit to the patient.
Okay, sir. It's clear. Thank you, sir.
Hello. Am I audible?
Yes, yes, you are.
Doctor, this was very good presentation. Sorry, Karthik, Dr. Karthik. I'm Rajesh. I represent a CRO. I'm the Chief Strategy Officer. I'll take that question and just add it. We are doing a couple of digital therapeutic trials, especially in the area of autism. And definitely there is a lot of, because the target audience was children, and this was played a very major role in bringing the compliance, a better compliance. And data was very useful, like you rightly presented in your paper. The compliance was close to 90 to 93% in the outcome. So the patient compliance, definitely it will be an interventional going forward. And we are getting a lot of, I can say, approaches in diabetes, in mental health, in child, and some of the GI related disorders, and it's picking up. And we have developed a lot of models for that, clinical research models for that. And going forward, this will be there in that.
Wonderful, wonderful. Very, very glad to hear that, sir. And also, I'm sure you will be following the new trend. I believe there are new artificial intelligence applications in clinical research, which is helping us to, I believe, optimize cost, especially when it comes to phase 2 and phase 3 programs, and also to accelerate clinical development as well. I do keep reading this, but since you are in the middle of the actual action, I'm sure some of these technologies will be very relevant there.
Yeah, because initially there were a lot of teething problems in designing a study, because end user were not used that there was a lot of convincing to be done, because patient consent was to be taken, and they were saying, I'm not taking anything, how do I get cured? So why should I use it? So a lot of convincing we have to do, and ICDs was supposed to be designed in a different way, and even the regulatory needs to be told, and definitely government side, regulatory side, everybody is looking at this as a positive step because the results are good.
and it is going to pick up. That's what I keep on talking to everyone, that digital therapeutics is going to come and it will be there. And practically we have been day in and day out, we are into that trials. Yes, there were initial teething problems, but we are able to fix it up, right? Like you said, that all the community within, like medicine, clinical research, the principal investigator, the patients, everybody has to be on a platform then to get this better benefit of this therapy.
Thank you. Very glad to know that. Absolutely.
Any questions?
A very good afternoon, sir. This is Dr. Siddharth Desai from Siddharth College of Pharmacy, Mudhol State. Sir, we heard during your delivery digital health insurance. Sir. Just please add us something more, sir. So as health workers, we can transfer the benefits to the public, sir. Digital health insurance, please.
Absolutely. I think as health workers, there are multiple technologies that can be leveraged. Of course, naturally, as you would realize, it will need endorsement or funding from a specific organization. I can talk about specific organizations that are working in this direction, a few of the pharma as well as tech organizations, especially the ones who are working on connected health. If we were to talk about community-based, naturally, under the NCD guidelines of the government, both the state government and the central government, there are specific NCD programs focusing on cancer, specific NCD programs focusing on diabetes, high blood pressure, and cholesterol. So these are broadly the 4 areas, and the 5th area is thyroid disorders, endocrine disorders. In each of these, there are specific digital health interventions, I would say, which involve, number one, monitoring on the field. As a health worker, when the health worker goes to the field, they can do live monitoring on the field of specific parameters related to sugar, blood pressure, cholesterol. In case of cancer, it is mostly about detection through pap smear or detection through minimally invasive or non-invasive mammography or breast cancer screening equipment that is available. A lot of these equipments are already artificial intelligence enabled, where it already provides to a great extent a close to accurate clinical impression. But at the end of the day, we have to understand that artificial intelligence is not a human intervention. Artificial intelligence is a machine learning algorithm which gives an impression. It is not medico-legally valid. It still has to be endorsed by a human doctor, a qualified doctor. But it fast tracks the whole thing. Okay, so there are applications which health workers can use, but the next step of what health worker can do is to motivate the patient to adopt and use what we call patient-reported outcome technology, where even when the health worker is not available with the patient himself or herself can monitor, do a self-monitoring of the health parameter, report it on an app, as simple as using WhatsApp or Signal or Telegram app. You can do self-monitoring and report it. It directly updates into their personal health record. The doctor comes to know, the nurse comes to know, the paramedic or the healthcare worker supporting the patient comes to know how the patient's health trend has been. So it is a joint work between the field-based health worker and involving the patient as a equal partner through patient-reported outcomes, both of which are, enabled through technology.
Thank you. Thank you.
Any more questions? Thank you, sir.
Thank you very much for the opportunity again.
Lunch has been arranged in the college quadrangle, so
Feedback. Yes,
There is an announcement. Just
I thank all the delegates for patience presence for this morning session. Now we are into end of this keynote session. You have a lunch, and after lunch, the scientific sessions will begin. There are slight changes in the scientific sessions because of want of a time. The first pharmacognition, pharmacovigilance, and risk management. which is supposed to be held in this Shivale auditorium. So only oral presentations belongs to this category will be conducted in this auditorium, whereas the poster belongs to the same category will be conducted in the Form B classroom 1, that is the ground floor itself. And drug delivery system, again lecture hall 1, as usual 18 presentations are there. So that will be conducted in the lecture hall 1. Whereas for modern pharmacochemistry and modern pharmacology, lecture hall 4 and lecture hall 3. So 1 to 10 will be organized in the lecture hall 4, whereas from 11 to 20 will be in lecture hall 3, that is in the first floor. So please plan accordingly, submit your presentation to the volunteers so that you can complete with all the scientific presentations. And we have a valedictory function at 3:45 to announce the winners of the scientific presentation. So please try to complete all this by 3:30 and be present by 3:45. Don't worry, we have only 30 minutes of valedictory function. I can understand you people have to go back to your places again. So from 3:45, we'll wind up by 4:15 and maximum 4:30. Thank you. Thank you. You can have your lunch. And please assure that you are back to the presentation rooms by 2 o'clock. Thank you.
Participation certificate will be issued at 4 p.m. in registration counter.
As you time given is 10 minutes. In 7 minutes, they have to complete the presentation. And in the remaining 1 minute, they have to go for concluding their presentation. And in the remaining 2 minutes, there will be a question and answer. Hope the things are clear. My name is Babu Bandeshetty. I work in the Faculty of Pharmaceutical Sciences at Adichunchanagiri. Today I am going to present the topic that is novel technologies and techniques to determine the protein structure. That is, which follows the structure-based drug discovery approach. So, what the plan is here? There are 3 slide pictures here. These are all the organic compounds where these pharmacists will synthesize, design, and everything. And here, these are the protein structure where our humans, we have millions of molecules. As we understand in genomics talk, the drop sequence of human genome is ready. That is, we have a sequence infrastructure, but we don't know the shapes of each and every molecule work in our body. So that is the first thing, we need to know the shapes and the 3D structures of all the proteins available for the, especially for the human genome. So second bottleneck is, and which fragment will block the activity of this particular protein. So the protein which is in zinc, that relates to cancer related protein. If you have a cancer, then we need to find which fragment, which ligand will find its activity. So major bottleneck is we need to experimental approach of this structure-based discovery is to crystallizing this protein with the suitable fragment. That is the crystallization step. So now this is the platform around the world to increase the speed and to make the process automated, that is generating the large amount of crystals and soak them with different types of fragments or ligands library and collect the diffraction data, X-ray diffraction data in high speed and determine its structure and find the which molecule is binding and which place.
Sorry.
So the lot of platforms have developed around the globe in all the places, and the current situation in the platform is very high throughput. That is about 250 fragments can be screened and collected at all the structures, and per week it will be around 1,000 to 2,000 structures will be solved, and the success rate where the fragments will bind to the structure is still very poor. So what I am going to try to show you here is how the throughput is increased. See, this is just the definition of the fragments. So here is, this is the impact of this method. It is time-consuming method, that is fragment-based or structure-based drug discovery. Is gave 4 drugs for the clinical use so far, and more than 40 molecules are in clinical trials. So this is very high impact. Okay, so now what procedure we follow is, that is strategy again for the high throughput ligand screening to determine the protein ligand complex crystals. 2 types. One is we can do the co-crystallization, that is adding the protein as well as the ligand and set up the trace for the crystallization, or soaking the fragments into the grown protein crystal in order to make the protein ligand complex. So now how we can do that in high throughput manner? Because we have a library of fragments, millions of fragments are here, and the proteins are there that should be grown in the crystallization form. So a robotic approach is called a mosquito robot, where it can grow several hundreds of
crystals in the uniform setup in the tray. And in the bottom, in another tray, we have a library of the fragments, and we are mixing this crystal with the fragment in this approach. And again, so taking the crystal and putting it to the facility, you can look at it, the facility, where this robot is picking the crystal from the drop and freezing in the liquid nitrogen and putting it into the diffractometer, where one can perform the X-ray diffraction experiment. So in principle, X-ray diffraction experiment is very simple. That is putting a crystal, frozen crystal into the beam and collecting the diffraction data. Once you have the data, it's like here, you can see the diffraction spots here. Here, the important concept is the resolution. If your diffraction crystal is diffracted to 2 Angstrom or 1 Angstrom means, you can see the each and everything, how the interactions, how the molecules are arranged at the atomic level. You can see here cysteine residue with the sulfur atom, you have a very high density, and you have a tryptophan here. So what I'm trying to tell you is that with this approach, you can see what is going on at the atomic level, the protein and ligand interactions. So the experiment, the idea is very simple, but experiment is very difficult and very expensive. See, this is the place where these platforms are located. This is the ring here. You can see the ring. This is the synchrotron ring, where the synchrotron radiation produces extremely bright light to this kind of experiment. This cost around $1 billion to make it. So with this synchrotron, we will have the high-speed electrons, X-rays are moving around the storage thing, and it will be ejected out in these lines here. So each and every line is called a beam line, and varieties of experiments can be conducted here. One among them is the protein crystallography experiment, where I work is the beam line BM14, that is in collaboration with the Department of Biotechnology India. And the facility here, where at the end station you get a beam here and sample is loaded here. With the help of this auto mounter, the samples are shipped from India by freezing, putting it in this loop here with these wires, and the frozen samples will sent to the synchrotron. And this is the closer look of the where the sample is loaded, and automated tools to carry out the data diffraction data collection. And here the 6-axis robot, and again the things are get highly automated, and these sample loaders are also miniaturized. This is where the sample is located. That will be micro size, and again lot of automation was developed to carry out this X-ray diffraction experiment at the synchrotron. And this is the beam and automatic software, and in collaboration with various synchrotrons. And we are putting a sample here at the beam and collecting the data. And as and when data collection is going on, at the same time the processing is also start. It is also automated, and the process data is automatically went to the data processing, structure refinement, and ligand fitting. And this is the last slide, where we will find the electron density for the ligand, that is a drug molecule. Okay, more or less everything is automated. See, this is the facility right now in Italy. This is the Department of Science and Technology is established. This is, I am the part of this development, and our sample, we can send the samples here to carry the experiments. Thank you. Thank you very much, sir. Sir, can you go to the title? Title process. Particular title, is it a kind of a review, or are you enlightened as the procedure of No, what I am trying to tell you is that this is automated technologies. I was involved in developing the technologies and also the novel techniques I have adopted for the X-ray diffraction data collection. And the facilities where I showed you, I was involved in developing automation and robots development and programming and data collection methods to solve the structures. And yeah, different methods. Only crystallographers will be able to do those kind of things. My question over it is not review article. It's a general facility for the Indian crystallographers. Can use it, including our Adichunchanagiri University, where you are giving us a sample. Where at the end we grow a protein crystal, we ship to this facility. That is what we are planning to do it here, and we are collecting the data by remotely connecting the facility. That is, remote data collection can be performed from here. Yes, facility, and is anyone else interested? All the participants from Bangalore and different colleges interested in giving us a sample for the protein crystallography experiments. Please contact me. Thank you.
All the proteins are crystallizable.
That is good question.
On the proteins itself. So whatever the protein we are going to use it for the studies, they should be available in the crystal form.
That is the bottleneck. That is why the robot is here. We don't know in what condition this protein is going to crystallize. So you have to screen millions of solvents and beds, buffer, and different concentration to check where will be my protein will be happy. So crystallizing a protein itself a bottleneck. That's why robot is increasing the number of experiments. Earlier, each drop you have to do it by your hand. Each time you have to, suppose if you have 96 drops, you have to pipette 96 times. Okay, so robot is doing at one time 96 drops. So the eye throughput is increased, and also you can screen millions of conditions to get the success rate of the crystallization. And also, second thing is, now you can set up 96 wells with 96 times pipetting, but in the next day, whether that drop is got the crystals or not, you have to watch through microscope. So now that is also automated. Automatic pictures will take by imaging technology. This is all we have developed at the synchrotron.
Preparing a crystal, the protein may lose its structure.
Yes.
Maybe it one of the different.
That's no, it can be purple, it can be precipitated, or it cannot be crystallized at all. So if you set it for 96 wells, all 96 wells will be gets precipitated, or 96 drops is going to be a clear. Success rate is very poor. That is a major bottleneck in X-ray crystallography. Especially the crystallizing these plant proteins are very easy. Humans protein is very difficult. That is like a high hanging fruit. Okay? In addition to that, membrane proteins again even difficult. So that's why this method gives you that, but you need to know where is the problem in your body. So these proteins work in your body. You should be able to know the shape of the molecule or protein to identify a drug for it. If you don't know how it is looking like, for example, coronavirus, you don't know how it is looking like. Okay, how do you identify the vaccine for it? You have to block its activity, right? So you have to do this technology, and you have to, this is the experimental evidence. I saw the people are looking at autotopina, Schrodinger. These are all just modeling work. It's not closer to the reality. This is closer to the reality. If you are able to crystallize a protein of your interest, okay, that is related to cancer causing the protein, so you will definitely identify a drug for it. Moment you know the shape of it, you will know the active site of it, and moment you know the active site is which substrate will bind to here or which ligand will block its activity, that is inhibitor. So identifying the inhibitor and knowing the structure is most important in the steps of drug delivery. Why do you think it will take 15 years to find a drug? Because of this. It's a slow process, and it's not straightforward.
Any question from the audience?
Yes, sir.
Thank you.
No, sir, not created, sir. Just I update BNS only, sir. Just I update BNS only, sir.
Good afternoon. Durga Shivakar from Department of Pharmacy, KD College of Pharmacy, Bengaluru. Today, I'm here to present
Yes, sir.
work, pharmacological and old pollution deterrence of a Western Ghats plant named Adichunchanagiri lagucha using 3 different species of mosquitoes.
It's a password. Yes, sir. Yes, sir.
So mosquitoes are the vectors that carry many diseases, they may be lethal or life-threatening also, so such as malaria, dengue, chikungunya, and so on. So in the daytime, nighttime, somehow we can avoid mosquitoes by using net or something. But in daytime, it is unknowingly the mosquito can come and bite, so it can carry many diseases which you don't know. So it is very important to control these mosquitoes in its larval or exterior layer. So we have many synthetic pesticides that are used to control these mosquitoes and larvae. Where those mosquitoes will develop resistance towards the synthetic pesticide, and also the synthetic pesticides causes the harmful effects on the organism that are not a target of that particular pesticide. So in our nature, we have enormous wealth of the plants in which bioactive constants that make and that have the properties to which can be altered this synthetic pesticide, which can be used as an altered one. So one such plant is Adichunchanagiri lagucha. It's a Western Ghats plant, belongs to family Moraceae, and this plant is also called as monkey jack, laguchi. In Kannada, it is called as echadaguri or vatehuri. So this plant is used in Ayurvedic and Yunani system of medicines because of its has many medicinal properties such as antibacterial, anti-helminthic, antioxidant, and insecticidal, etc. So because of the insecticidal property of this particular plant, we are using in our study. So aim is to perform the pharmacological and oil pollution deterrence activity of leaves of Adichunchanagiri lagucha. Primary objective is to determine the oil pollution deterrence activity, and secondary is to standardize the leaves of Adichunchanagiri lagucha. Plant were collected from the Western Ghats of CC Karnataka in the northeast altitude, and was authenticated by the plant taxonomist named Dr. Santan P. from National Remedies Private Limited. Pharmacological evaluations such as moisture content, ash, total ash, water soluble ash, acid insoluble ash, different extractive values were carried out as per the standard procedure. So the next, the plant were extracted from the solvents by increasing polarity, that is petroleum ether, chloroform, ethanol. The mosquitoes of 3 different species have been used in this study, such as Anopheles cephasi, Culex quinquefasciatus, then Aedes aegypti, and these are collected from in and around of the Devanahalli and Yelahanka region of Bangalore. Before this, they had conducted this study on the lab-bred mosquitoes. Oil pollution deterrence is a process in which it expels the eggs from the detaching from the oviduct or preventing the laying of the eggs. So this was what were the mosquitoes which are undergone this oil pollution deterrent activity. So that egg which is laid by that, it has the low hatchability or viability. So this procedure was carried by under WHO guidelines by taking 15 rather female mosquitoes of 10 days old and 4 days after breastfeeding, and this was taken in a small cages, and controlled water was taken as a tap water, and different solutions. This solutions were taken with a different concentration from 50 to 500 ppm concentration, and it was placed in opposite side of the control one. Then this experiment was conducted in triplet and with the ambient temperature 27 degrees Celsius plus or minus 2, and 70 to 80% of humidity, and
light and dark cycle of 12 hours. After 24 hours, this which is laid by the control as well as the treated group were collected and recorded and this is calculated as percentage effective repellence. Or pharmacological evaluation result were found to be nature of the leaf of Atrocarpus lacucha was found to be oblong, ovate, broad base, acute effects with short petiole, and color of dark shiny outer surface and pubescent inner surface. Odor of characteristic odor, taste of slightly bitter. Texture was rough and fibrous. And powder microscopy was conducted in which the characteristics was found to be midrib xylem vessels, annular thickening vessels, spongy mesosphere, glandular trichomes with brown head, long covering trichomes with pitted vessels and lignified fibers. Coming to physiochemical properties, this was the test were conducted in which a moisture content was got of 7.543 with and rural ash of 4.071, water soluble ash with 6.688, and acid insoluble ash with 6.351. Foreign organic matter with 0.2 and extractive values were conducted in 3 different solvents: petroleum ether extract, soluble chloroform soluble extract, and ethanol soluble extract. Their result was found to be like this. And phytochemical screening was performed in which petroleum ether extract has shown presence of alkaloid, phytosterols, and terpenoids. In chloroform and ethanol, phytosterols were found. So this analysis was conducted to know how in which solvent or solutions the plant is giving its fluorescence. So these reagents and solvents were used such as methanol, methanolic NaOH, ethanolic NaOH, as powder as such with the water and so on. So this was observed under white light, 254 nanometer, 360 nanometer, and these were the results found. So oviposition deterrence activity was done with 3 different extracts of leaves of Atrocarpus lacucha, one with petroleum ether and chloroform extract, and lastly ethanol extract, in which chloroform extract has not shown any repellent activity, whereas petroleum ether and ethanol extract have been shown. So in as is in the as you see here, with Anaphyllus depensi species have shown 104.95 repellents with Culex of 130 and Aedes of 1 to 14. This is a petroleum ether extract result. And lastly, ethanol extract in which 226 for Anaphyllus, 274 for Culex, and Aedes we got of the 86. So as we can see here, Anaphyllus has got a little sensitive than this Culex and Aedes species. So I would like to conclude by saying that petroleum ether extract have shown the effective repellence activity than the ethanol. The order of oviposition deterrence activity is found to be petroleum ether is greater than ethanol, ethanol is greater than chloroform. I would like to acknowledge my college, Kelly College of Pharmacy, Bengaluru, my guide, Rajama AJ. Thank you all for this opportunity. Thank you, sir.
Question is the selection of the solvent for your leaf for your plant.
Yes.
Both the plant or maybe part of
leaves.
What was the reason?
Sir, because of the screening purpose, we have used the different solvents, petroleum ether, ethanol, and chloroform. As the polarity increases, the different compounds will go in that particular solvent. So we want to try with those particular solvents.
The concept of like solvent likes dislikes. So on the basis of that, you mean to say that your leaves has contained only the polar constituents?
No, sir, we want to check it with those solvents.
You got the satisfactory result.
So you told that you collected the mosquitoes from the nearby area plant.
Yes, sir.
So how do you identify those 3 different species from those mosquitoes?
Sir, specialists, they have your the green mosquitoes.
So after that, you said that petroleum ether extract.
Yes, sir.
I see the petroleum ether extract normally contains, if you extract, highly non-polar compounds are extracted out.
Yes, sir.
Alkaloids are medium polar compounds. But is shown in petroleum ether extract, alkaloids are present.
Yes, sir. They have got the result.
Is it possible? Because alkaloids normally, if it's in the form of salt, it will be in the polar solvent. If it is in the form of the free base, it will be normally found in chloroform or dichloromethyl. It will not there in the petroleum ether or NX and whatever it may be, highly non-porous.
Yes, sir.
The result shows that it is present in petroleum ether extract.
I do see.
Yeah.
I check it.
Then even if you see the results, you say that petroleum ether extract is showing very good activity, followed by that is the ethanolic extract. But in between this is the chloroform.
Yes,
If petroleum ether shows the activity because it's a successive extraction, right?
Yes.
So after you use the same plant material using the petroleum ether, then use the same plant material for chloroform. Then after that you are going to use ethanol. Then the plant material is like if compounds are present, it might be extracted out by chloroform.
But it is not showing any activity.
But that's what I'm asking. See, once it might be the polar, whatever the component is there, you're saying petroleum ether is showing good activity, other compounds have been extracted out by the petroleum ether. So then chloroform comes next. So there's nothing is there. Though it contains the phytosterol, it's shown phytosterols are present in all the three solvents. Extract. But ethanol show you shown only phytosterol is there. The same phytosterol also there in the chloroform.
Yes,
and ethanol when shows the activity even the chloroform.
So in what I have got the graph, in this they have taken below 2000 ppm. In chloroform, they have got more than 2000. Whichever we take the results, we prefer in lower concentration. So this petroleum ether, given the its result or effective repellence in lesser quantity. So they have chosen as its effective. It is effective than other.
So what was the solvent vehicle used to dissolve this extraction? You cannot use the same chloroform or the petroleum ether. Once you get the extract, you have to dissolve it in some solvent, then you have to keep it in the cage, you said that, correct? For the feeding purpose.
Yes, sir.
What was the solvent used for the feeding?
Actually, I'm not on that, sir. Seniors work, they have done this. Just I'm doing the presentation. Under my guidance of my director.
Thank you.
Thank you, sir.
Name? Name? B.
A very good afternoon to another year. Myself, Dr. Priya Shetty, faculty of ICIAPS Dr. Prabhakar Kure Basic Science Research Center, and I'm working as a research associate. And basically, I'm from faculty of pharmacy. To begin with, the oral presentation of my title is the title for my oral presentation is Epigenetic Nanoliposome Formulation and Quantitative Estimation of Epigenetic by Validated HPLC. Introduction is initially epigenetic is an isolated compound from chamomile plant, and it has been selected from chamomile plant belonging to family Asteraceae, and it is mostly widely used as medicinal herb in China. And the scientific name of this plant is Maticula chamomile. And it is almost present with all the vegetables and fruits. In vegetables, it is present in carrot, onion, cilantro, and all the green leafy vegetable. And in fruits, it is almost present in citrus fruits and also in tomato. And the chemical structure of epigenetic is 4,5,7 trihydroxyflavonoid with wide range of biological activities, including antioxidant, antiviral, antibacterial, anti-inflammatory, chemopreventive, and even also in cancer. Literature survey revealed that there was no study on HPTLC. Hence, the study was taken to develop the HPTLC method for epigenin in bulk and its nano formulation. Coming to the need of study, it is a part of the PhD project wherein the study was carried out for breast cancer. So, this is the basic need of the study. Breast cancer infection in the world is the second cause of death. According to WHO, in 2022, total 6,26,679 people surrendered to disease. Actually, development of therapeutic resistance and metastasis is a major challenge with breast cancer therapy. chemotherapy and radiation therapy have severe adverse effects and drug-resistant disease recurrence. So, a class of natural flavonoid named epigenin plays a major role in prevention of cancer, but it has got limitations like poor viability and low water solubility. Hence, there was a need to overcome this limitation and improve the targeted specificity. So, this was designed and developed, that is epigenin nanoliposome. Coming to the HPLC, it enables the most complicated separation, identification, characterization, and for qualitative and quantitative analysis. The aim of the study is to investigate the effect of epigenin nanoliposome in breast cancer. Objectives are to formulate epigenin nanoliposome via ethanol injection method and to quantify the epigenin by developed validated HPLC technique. Coming to the materials and method, epigenin was a gift sample of actin chemical China. HSPC, which is a lipid, was also gift sample from Nippon, Germany. The solvents used for HPLC were methanol, toluene, ethyl acetic acid, which was procured from SD Chemicals, India, and silica gel aluminum plate was procured from Sigma Aldrich, and all the chemicals were procured from Central Stores of Basic Science Research Center, Belagavi, and from Kelly's College of Pharmacy. Coming to the free formulation study, initially pre-formulation study was done by taking all the analytical methods like UV, HP, but here we are focused on HPLC. Preparation of nanoliposome was done by ethanol injection method, and HPLC method development validation was done for epigenin. Finally, it was quantified. That is, quantification of epigenin was done in epigenin nanoliposome. Coming to the result and discussion, here the chromatographic condition, that is method development, was done by taking a mobile phase, toluene, ethyl acetate, and acetic acid in a ratio 6:3:1 volume by volume on a stationary phase, that is HPLC silica gel plate with size of 20x10. And the UV scan wavelength was at 269. Nitrogen gas pressure was used for application of sample. And coming to the characterization of this nanoliposome, the particle size was within the range, that is 36 nanometer. PDI was 0.27, zeta potential was -137, and percent entrapment efficacy of drug was 73%. The linearity range was within 10 to 50 nanogram per band. And also based on this calibration curve, the R-square value was 0.998, wherein the LOD was 2.44 and LOQ was 7.0, and this was the as the concentration increased, that is 10 to 50, even the band size also increased. Graphically, it was represented like this. Coming to the precision parameters, here the precision parameter was carried out for intraday and interday. Intraday is nothing but from morning, afternoon, evening per day, and interday is day 1, day 2, day 3. Wherein one concentration, that is the middle concentration, was taken and applied. The samples were applied in triplicates, where the RSD was observed within the acceptable criteria, which was less than 2. And coming to the specificity, after carrying out the specific parameter, it was found that epigenin standard and the epigenin nanoliposome had similar RF value and were both overlapping. The chromatogram of this epigenin and its nanoliposome was shown in figure, and the RF value was round about 0.45. Coming to the robustness, here in robustness, usually we select different concentration of mobile phase and volume of there was change in volume of mobile phase, and even the duration, saturation for duration of chamber. That is, initially all were within the acceptable range. That is, the criteria, all the readings were within the acceptable range, and the criteria was fulfilled because it was less than 2%. And for quantification, the triplicates of the sample, that is epigenin nanoliposome, was applied on the silica gel plate. And the data is presented in table 5, and the mean and standard deviation of the result was obtained and calculated. Here for standard, we had taken 1 mg per ml, and for sample, that is nanoliposome, we have taken 4 mg per ml. Based on that, we quantified 306 as the result. Coming to the summary, here the mobile phase used was toluene, ethyl acetic acid in a ratio of 6:3:1, and the stationary phase was transparent and was good evidence by this. The newly proposed technique was validated for parameters which include linearity, range, LOD, LOQ, precision, robustness based on ICIAPS-R2 guidelines. And the linearity of epigenin was ranged from 10 to 50 nanogram per band, wherein the LOD was 2.44 and LOQ was 7.40. The precision and data finding showed newly developed method was accurate since the percent RSD for the peak area was within the intraday was determined as 0.9% and interday was 0.52%, both which meet the criteria below 2%. The determination of concentration of standard drug present in nanoliposome were quantified in triplicates, and the nanoliposome were determined by having a concentration of 306.5 microgram per ml. To conclude, HPTLC was developed and validated based on ICH guidelines, and it was discovered that it was a straightforward, linear, precise, specific, robust. The epigenetic nanoliposome drug delivery system can be analyzed using the described approach. Future prospects are quality control for finished products, impurity profiling, batch-to-batch validation, stability study for nanoliposome and inhalable liposome. Acknowledgement and references.
This one, I think, we talked about the epigenin. This epigenin means whatever the study we did it only for epigenin or nanoliposome epigenin.
Sir, actually, epigenin is an isolated compound from active chemical chain, and it has got drawbacks like poor viability, low water solubility. And we have prepared a nanoliposome from taking this drug. We have added different excipient like polymer, lipid, cholesterol, and then we have prepared a nanoliposome with the ethanol injection method. And this epigenin nanoliposome is being quantified in HPTLC analysis.
RF value of this epigenin
0.45, sir. It was matching. The standard and the nano formulation were almost matching, sir, the bands. It was 0.45.
When you prepare, when you add lipids to this,
yes, sir.
The RF value will not be changed.
That is what I'm telling. Actually, standard is taken, that is that drug epigenin was 1 mg per ml. The sample what we take, that is epigenin nanoliposome, is 4 mg per ml. Diluted.
When you see that you are converting into the soluble form, right?
Yes, sir.
But you're using the same silica gel plate.
Sir, actually, this is not taken in liquid form. We had lyophilized that and we have taken the powder.
But
Both
you
the
have
powder.
to dissolve it in some solvent.
Methanol, sir. Methanol.
But that's what I just not confused because you're converting it from the lipids and you're applying. And there will be, when you use the silica gel stationary phase, for the polar, we are going to have the different stationary phase, and non-polar, we are going to have different stationary phase. But we are working in the, we are using the same plate for both these two. The nanoliposomes as well as epigenin also.
Yes.
Is it possible?
Yes, sir, it is possible. So only we had quantified that. No, actually, the drug also is dissolved in methanol base, and the formulation also is completely dissolved in methanol. Then it is centrifuged, the supernatant is collected, and then it is applied, sir, on the plate. There was no much difference, sir.
So you're telling it's a new method. New method
Yes,
means,
sir.
what is the preparation
Actually,
of the nanoliposome is a new method, or the
quantification is new method, sir. Actually, in nanoliposome also, for liposome preparation, there are various methods like thin film hydration, reverse phase, ethanol injection. 99% of literature have only thin film hydration for preparation of liposome, but I have used ethanol injection method.
And how do you differentiate the nanoparticles with the nanoliposome of epigenin?
So nanoparticle actually is a whole thing, like when you do preparation, and nanoliposome is like, there are various, based on the structure it is identified. So liposome is usually spherical and oval in shape, cubosome is cube shape, and ethosome is, it is almost loaded with ethanol and lipids. So the
structure of similarity seems to be like a query.
Yes, sir, circle, oval shape, sir. Based on cell and TEM analysis, we get those pictures.
No, my question was like, when I saw the structure, epigenin,
yes,
it was almost 90%, it was resembling a covering structure. See,
sir, that is isolated compound structure.
I agree with you. Why have you made any attempt to go for synthesis in that molecule? This needs to be like almost like covering molecule. We prepared a lot of derivatives of covering molecule. This molecule is also prepared only one OH group is explained this.
No, sir, I've not tried it. Thank you, sir. Good afternoon. Myself, Dr. Pandaresh, and I'm a faculty of Adichunchanagiri University, the School of Natural Sciences. So today here I'm presenting a work which was carried out in US during my postdoctoral time. So this work speaks about the supplementation of walnuts in the diet and the progression of Alzheimer's disease, which was studied in the transgenic mice model. Okay, so here what we have done is, so you know that Alzheimer's disease is a age-related progressive neurodegenerative diseases. If anybody, there are numerous number of hypotheses have been proposed. So the major thing, if anyone asks why Alzheimer's disease is caused, they say mainly it is because of the amyloid beta protein or aggregation of tau protein. So here I am mainly focusing on the accumulation of amyloid beta and the clearance of the amyloid beta in the brain samples of transgenic mice. So this is amyloid precursor protein. This is a transmembrane protein. This is the one which is responsible for the segregation of amyloid beta. This is the N-terminal end, and this is the C-terminal end, and this is the amyloid beta domain. So this protein will be processed by secretase enzyme. It may be alpha, beta, or gamma secretase. Alpha secretase will cleave in between the amyloid beta domain, whereas beta secretase will cleave at the terminal end. Gamma secretase will cleave at the C-terminal end. If the protein is being processed by alpha secretase and gamma secretase, it will give rise to P3 protein. The function of this P3 protein is exactly it is not known because it will be greatly degraded and it will be used for some other process. And AICD will be there. That is nothing but APP, intracellular domain. It is involved in cell proliferation, cell differentiation, and other process. And this is the C-terminal fragment, and this is the soluble APP alpha. Even this will protect the brain from excitotoxicity, and it is involved in cytoprotection mainly. So this is the non-amidogenic pathway. So when coming to the amidogenic pathway, instead of alpha secretase, it's the beta secretase and the gamma secretase will be cleaving. So there will be release of amyloid beta. So the monomer form of this amyloid beta is non-toxic. But when this amyloid beta, it gets aggregated, when it gets oligomerized, that is toxic. It forms the lesions, that is the extracellular lesions. It is the one which cause the damage. So here in this study, we are mainly focusing on 4 different things. One, the enzymes which are involved in the processing of this amyloid precursor protein. The second one, the enzymes which are involved in the degradation of this amyloid beta protein, which is already existing. And the cholinergic markers, because the acetylcholine or the cholinergic neurons are the major things which will be affected during the Alzheimer's disease. So we are focusing on the cholinergic markers, mainly the enzyme which is involved in the synthesis of acetylcholine and the degradation of acetylcholine. That is acetylcholine esterase and acetylcholine esterase transferase respectively. And the neurotropic factor, so which is overall involved in the functioning of the brain, normal functioning of the brain. So here we have used this is a transgenic mice model. It is a Swedish double-mutant mice, which we have used here is TG2576. It's a model that we have used here. So here what has happened is in the amyloid precursor protein we have 3 different isoforms. So in that isoform 659, so that we has been mutated at 2 positions, at the 670 position and 671 position, where K has been replaced with N amino acid and M amino acid has been replaced with the N. That is lysine with arginine and methionine with leucine. Okay, and we have supplemented the animal with 6% and 9% walnut diet. And we have estimated its efficacy at different age points, that is 4 months, 9 months, 14 months, and 19 months. So this is age dependent study that we have done with, this is about the animal grouping. So with the different age points we have studied starting from 4 months to 9 months. So each group had a control group, wild type animals as well as transgenic animals. In transgenic animals, we had fed the transgenic animals with the control diet, with the 6% walnut diet and with the 9% walnut diet. So the experiment was started at the 4th month. So we don't have 6 months and 9 months in case of 4th month. So this is the data. So this is about the alpha secretase, beta secretase, and gamma secretase which are involved in the processing of amyloid precursor protein. So if you see the data, this is a wild type mice, and the TO. TO is nothing, T0 is nothing but these are the transgenic mice. T6 is nothing but transgenic mice which are fed with 6% walnut diet. T9 is nothing but transgenic mice which is fed with 9% walnut diet. Similarly, it goes, And if you see here, so there is a age-dependent decline in the alpha secretase activity. Okay, and there is a age-dependent increase in case of you see T0 here, T0 with each and every group. So age-dependent decline in the alpha secretase, age-dependent increase in the beta secretase as well as gamma secretase, which clearly says that so the trend is more towards amylogenic pathway where the amyloid beta is being secreted in the animal. So with the supplementation of walnuts, we can see that so the accumulation is not that much. The secretase activities was restored as well as the beta secretase activity was almost near to the wild type. If you compare with 6 months, 9 months, 14 months, and 9 months. So this clearly says that the walnut supplementation at 6 months and 9 months has protected the amyloid precursor protein from the cleavage. So next, we are focusing on the two different enzymes, that is neprilysin and incilysin. These are the two enzymes which are involved in the degradation of secreted amyloid beta. Okay, so this is a endopeptidases and this is a metalloproteases. This is a zinc-dependent metalloproteases which gives the metalloproteins. So you know that amyloid beta is also a protein, right? It's a peptide. So even that will be degraded by these two enzymes. So here also, if you see that there was no significant age-related decline was not observed here. But yeah, if you compare with the wild type, so in the transgenic animals, the level of this neprilysin as well as incilysin were depleted significantly. Okay, with the walnut supplementation, both at 6% as well as 9%, it was restored. So now coming to the acetylcholine esterase, the enzyme which is involved in the degradation of acetylcholine to acetyl and choline, and the choline ester transferase is involved in the space of acetylcholine. So here you can see there is an increase in case of transgenic animal, which has come back to normal in case of supplementation of walnut. Here there was an increase was there, which has come back to the normal condition. So similarly, the neurotrophic factor, that is BDNF, that is brain-derived neurotrophic factor, also we have evaluated. So where we have clearly seen that the expression or the level of this BDNF was completely normalized with supplementation of walnut. So conclusion, what I want to say here is, so if you see the results clearly, it clearly shows that the short-term supplementation of walnut for a very shorter duration is not having significant effect. If you have seen that results very properly at the 9 months, the data was not that significant. But if you are consuming walnut for a very long period, for very long duration, then definitely it has a potential, okay, to treat against Alzheimer's disease. So here we are targeting the enzymes which are involved in the secretion or generation of amyloid beta. If already the amyloid beta has been generated, we are targeting the enzymes which are degrading that amyloid beta, and we are also focusing on the cholinergic markers as well as the neurotrophic markers. So this is the reference which has been already published. A part of work is already published, and this is my acknowledgement. I thank to the New York State Institute for Basic Research for funding, and it was mainly funded by the California Walnut Commission. They had funded more than $1.2 billion for this project. So it was a huge study where we have done, we have gone up to the transcriptomic and epigenomic level. Since the time of shortage, I have not that data here. So that's it. Thank you.
Yes, sir.
Whether this walnuts reported to have this activity against Alzheimer's.
That's what we wanted to check. Actually, walnut commission, many people says, the shape of walnuts, you see, it is more similar to brain. So they say there is some correlation between brain and walnuts and all those things. Yeah, if you see the individual component wise, it is very rich in omega-3 fatty acids, and it is having a lot of antioxidants and XYZ, so and so. But we are not much worried about those things. Only the thing is we are worried. What we wanted to say is, see, if you are isolating any active component from a walnut, or if you are giving it in the form of medicine or anything, the people will hesitate to consume, to take it. But if you give in the form of food, it is a form of you are not mixing anything here. It is just a pure walnut. So we have not extracted anything. We have not isolated any component. Nothing. This is a just walnut supplementation in the diet study, where we are supplemented with 6% and 9%, which is equivalent to 1 ounce and 1.5 ounce, which is as per the standard regulatory bodies what they had earlier. So here we are just supplementing the walnuts, and we are checking the efficacy of the enzymes which are involved in the Alzheimer's disease. Yeah, and yes, for the shorter duration, it didn't show any significant effect. There was no effect at all. But if you are feeding the animal for a very longer duration, then there was a significant effect of this. Not only in terms of processing the amyloid beta segregation, in also in terms of clearing the amyloid beta which is already segregated in the brain or any other tissues, any other regions. And it is also modifying some other protein markers, RAGE and LRP, lipoprotein receptor related protein 1. So there is one protein which is involved in the export or transport of amyloid beta from brain to the circulation, where the amyloid beta, because now the time has gone, where people were seeing the accumulation of amyloid beta only in the brain, where it is to cause extracellular lesions. But we are also looking for aggregation of amyloid beta in other tissues, mainly in liver, kidney, and other tissues like that.
Regarding the dry fruit content and its activity, many ancient books might have given the information. They are much useful in the scientific system. Some of the dry fruits are very much useful in releasing of dopamine. When all such contents are already present, then with reference to that and this work, how it differs?
Sir, here we are mainly targeting first thing what we are the way of approach. If you have seen, we are mainly focusing on the enzymes which are breaking the amyloid precursor protein. Right? No, I am not speaking about the acetylcholinestase, sir. I am speaking about the alpha secretase, beta secretase, and the gamma secretase. And I am speaking about the neprilysin and insulinsin. If you see any paper, you will not even get a single paper. It says that the neprilysin or insulinsin is involved in the, neprilysin. Yeah, neprilysin. There are some targets recently, 2020 or 2019. At the end of 2019 and 2020, there are some drugs which are neprilysin based one. But insulinsin, no. There are no as such reports and no as such papers which have come to the which have been published or done. And another one thing is here what we are seeing, we are doing a holistic study. We are studying with the oxidative stress, because this study, whatever we have done here, right, it is just with the enzyme activity, with the translation, after translation, whatever the protein comes, right, the protein activity I have shown here. So in addition to this, we have also done at the gene level. Okay, gene expression also we have done. In addition to that, we are also checking for the post-translational modification. Once the protein has been translated, what actually the post-translation modification which has exactly happened. So even that also we have studied. And that post-translation modification, we are correlating that with the epigenetic study. That is, the gene, if your gene have to be expressed in the form of a protein, it have to undergo transcription and then translation, right? So the promoter region is the one which is the hotspot. So we are seeing the changes, that is epigenetic changes which are happening at the promoter region also. So that is in-depth study. So it is a very short duration which was given for me here for only for 10 minutes or 7 or 8 minutes.
So if I keep on telling that, it will be very difficult, or it will take time for me to introduce what I want to say. So for that purpose, as a holistic thing, I just gave the activity. That's it.
Good afternoon, everyone. I'm Jayashree from the intern. Good afternoon, everyone. I'm Jayashree, from the intern in San Jose College of Pharmacy, and I'm very glad to share this topic in front of all of you. So today I'm going to discuss about psychiatric morbidity and its correlations in diabetic patients in a tertiary care hospital. Because diabetes is generally a chronic illness, chronic disease. It's not just mean it's just chronic disease, so it's diabetes. But due to some psychiatric illness and all, this diabetic disease is becoming worsen and worsen. What is diabetes? Due to impaired blood glucose level, that diabetes may happen, and that too because of any insulin resistance or decrease in the insulin production, the diabetes and all will happen. That may be differentiated to type 1 and type 2 diabetes. And here the coexistence of psychological symptoms and this diabetes becomes a major challenge for the clinicians, as because these both are psychiatric illness and diabetes both leads to worsen the condition of the patient outcomes. So here we have to see, and depression or anxiety is one of the raising cause of the seeking healthcare. Presence of psychiatric illness seriously impairs the health-related quality of life among the diabetic patients. In diabetic patients, actually they take the medicines and all. For that it will be okay. In case of diabetic patients who's having any psychiatric illness also means it will lead to the outcomes of the patient will be somewhat worsened. And here, how it relates means psychiatric illness may lead to some neurohormonal changes in the brain, because in brain also glucose is needed. There in hypothalamic pituitary axis, it will act and it will release some sympathoadrenal hormones. Due to that, the people may anxiety or depressed, like behavioral changes also will happen. And psychiatric symptoms can decrease the adherence and also some of self-care problems, that is physical activity. These are not will be reduced due to the psychiatric symptoms. And individuals with diabetes are having 2 to 4 times greater chances of risk of psychological illness when compared to individuals without diabetes. So psychological burden of being ill is being triggered the psychiatric symptoms. So being diabetic also, they feel somewhat trigger on these symptoms. And what is aim is to study the prevalence. That means how much the psychiatric morbidity is relating with the diabetic patients, and what are the object means to estimate the prevalence of psychiatric illness in diabetic patients. In how many diabetic patients, whether they are suffering with any psychiatric illness or how many, like that. And we are comparing by taking the medication. Some people are taking insulin and some people will taking oral hypoglycemics. So we are comparing between those patients also. And here we are also studying the family dysfunction, because psychiatric illness may also, due to some family dysfunction also, it may cause. So we have to check whether because of that family dysfunction they are getting psychiatric illness or due to diabetes only. So for that and all we are checking that, and which gender is getting more psychiatric illness, the patients who are having diabetes. And materials methods, this is be done in SWIMS hospital in endocrinology ward, and we took 150 patients, and study materials we use this patient demographic proforma, which is having the patient age, what they are doing, their educational status, and they whether they are taking insulin or oral hypoglycemics, like that. Everything will be collected and was collected, and mini version scale. By means to assess the depression and anxiety, we use mini and APGR for the family functionality scale. So inclusion criteria, we include all the diabetic patients, those who are interested, and both either gender we included. In the same way, those who are not taking any psychiatric medicines, they only be included. And exclusion, we excluded the patients, those who are not interested in the study, and in the same way, those who are already having psychiatric illness, those we excluded, because we can't estimate whether it is due to diabetes or some other reasons. And we also excluded the patients, those who are having other complications like hypertension. These are the patients, only patients with diabetes only we took. And this is our results and discussion. And here we took 150 patients. Out of 150 patients, oral hypoglycemics took 118 patients, and insulin were taking 16 patients, and 16 patients were taking both insulin and oral hypoglycemics. And gender, more female are there when compared to male. And educational status is mostly the people are unskilled or illiterate people only. Out of 150 patients. Yeah, this is the oral hypoglycemic and insulin, how many they are taking. And this is the APGR scale. So here in this scale, they are giving the score according to the whether the subject or the patient is mingling with the family, or he is a introvert, or he is conversing everything. If any problem comes to him, whether he is sharing with the family or not. Through that, we can assess the psychiatric mentality of the patient. So that's why we assess. But we got normal only. 22 patients, 22% is in the range of 8 to 9, which is a normal. And next, here we found that depression patients are above the age of 40, because we included, all ages are included. So that too we found the age greater than 45 years were found. And in the same way, females are also having more psychiatric illness in the diabetic patients. And the person who are having low socioeconomic, we done it with the Kupuswamy scale, and the person who are having low socioeconomic, they can't afford to buy insulin and all. So because of that also they affected. Next, the patients who are having overweighted, they also, because obesity, if they are having obesity, obviously the patient might have get distress or some psychological illness. So those patients are having. And patients who are receiving, we found that patients who are receiving insulin injections, they are found to be more anxiety, because in insulin resistance cases and all, when we are taking insulin, in our brain, the cells get activated, and the hormonal changes will happen. Due to that, sympathopaternal neurotransmitter will release. So because of that, the patient may excite, and the patient may have some behavioral changes also. So that's it. And out of 88 female patients, we found 52 were found to be anxiety patients, and 15 patients were found to be depressed. In case of 62 male patients, 34 were found to be anxiety, and 11 patients were found to be depressed. So this is a chart. So this is the what I said. The patients who are taking insulin are 75%. That is 12 patients. Total are 16 patients. Out of 16 patients, 12 patients are with anxiety. In case of depression, also 5 out of 16, 5 are with depression, and 14 are without depression. And we gone through this through statistical analysis. So this is, we got significant level in case of anxiety. That is nothing but our study is related the diabetic patients, those who are diabetic, they are slightly having the anxiety problems also when compared to depression. You may ask, there are so many psychological illness, why anxiety and depression? Because so many articles we had referred and we have seen, mostly the diabetic patients, that too when compared to hypertensive, diabetic patients are mostly effective because of this psychiatric illness, that too depression and anxiety. So that's why we took this as a baseline study. And this is our discussion. In our study, we found 54.2% were found to be anxiety, and 16% were depressive patients. So,
how did you select? You were saying that most of the patients you selected are unskilled and ill-treated.
Yes.
Illiterate, sorry.
Yes.
Illiterate. Means
because they didn't study. We asked you, sir, whether you studied or not. I said no, sir. So I'm having, sir, but I'm having, sir. I'll show.
This work or someone's work?
It's ours work only, sir.
No, published this
Yeah,
work?
yes, sir. We are on the way to publication, sir. It is.
Then what are the parameters you applied to find out the psychiatric the nature of the patient?
That is the mini scale we used, mini depression and anxiety, we took that, sir, and APGR scale we used. Sir.
This diabetics like either of diabetic 1 patient, diabetic 2, or the
Yes, sir.
sometimes even the ladies, pregnant ladies also have.
Yes.
Which category of patients is selected?
Sir,
Majorly out of that.
Told in here we excluded the pregnant and lactating women. We excluded, sir. Excluded, sir, because through pregnant and lactating also some psychiatric problems will come, so we excluded that, sir. Thank you, sir.
Good afternoon to everybody. I'm Sumit Kumar, presently working as an assistant professor in Central College of Pharmacy, and I'm also a research scholar in Jayanti University, Karnataka. So today I'm going to discuss regarding the impact of predisposing factors in of ADRs in mostly in cancer chemotherapy patients. So here we know that incidence of ADR and occurrence of ADR will be the multifactorial, and it will be depends on many factors like age, gender, and their comorbid conditions and their priority conditions and their performance of what is the disease progression conditions and all. And here most of the health scenarios we are failing that to estimate or to evaluate in which patients most of the ADRs are occurring. So I am trying here to, we tried some scales which is to assess the incidence of ADRs and all, occurrence of ADR with the help of some other scales. So here most of the leading cause of the death in the world will be the cancer, and we are giving mostly chemotherapy, radiotherapy, and surgical oncology also. And after we are giving the chemotherapy, the patients will be suffered with either chemotherapy-induced toxicity and adverse drug reactions, and there will be the question of patient safety. So as we are learning towards the patient-centric health sector economy, the patient safety and the patient satisfaction is more important. If we are giving the chemotherapy, the cancer has been cured, but apart from that, the patient is getting more consequences from the chemotherapy, that is worthless. So here, if you could able to assess the predisposing factors in the patient, and we could able to find out who are the patients having more chances of getting ADR, and we are giving any prophylaxis for those patients, we can minimize the risk of hospital burden, and as well as we can improve the patient quality of life. So these are the objectives. We are here assessing the patient-specific parameters like age, gender, body mass index, and metastasis, whether the cancer is a METS or a normal benign cancer, and the treatment options, whether monotherapy and combination therapy. And we are using three scales here. One is a comorbidity impact based on Charlson comorbidity index, and a fatality index, and ECOG performance status scales. So this is a hospital-based study, and we have been undergoing who are the chemotherapy patients only we have been taken here. And these are the materials we have been used, patient data collection form and a Charlson comorbidity index, and Eastern Cooperative Oncology Group performance status, and a fatality index for the elders, and a Narendra adverse drug reactions for to assess the causality of the adverse drug reactions. So, and this is presently we have been carried out this study with 112 patients who are undergoing chemotherapy, and we have taken the incidence of multiple ADR as an indicator for the predisposing factors to be occurred. And first of all, we have taken the age as a parameter. The patients who are having more than 60 years, that means around 52%, we can see here, they are having more incidence of ADRs, and it was supported by one of our previous study, which has been carried out by George et.al. So they have also concluded that the advancing the age will lead into the more incidence of ADRs. And the next we have taken gender. When we are comparing, the males are having more chances of occurring ADRs when compared to women. That is 32% is having the women. And next one we have been taken is an body mass index. So the patients who are having underweight and obese patients, underweight is a 50% patients and obese patient is 47%. They are having more chances of getting in the occurring the areas when compared to the normal patients. And the patients who are having metastatic cancer is having more chances of getting area when compared to the benign cancer. So next one, treatment, who are usually this we know actually, if you are giving a combination therapy, the efficacy also will be more, and in the same time we may get the more sort of risk also. So like in the chances of ADR's occurrence. So that here, but in our study we have been seeing a similar type only. But when we are comparing the previous studies on the, there is a combination therapy is having more chances of getting an ADR. And the next one, frailty score. Frailty score will be used. This is a frailty score and ECOG performance status and the Charles comorbidity index will be used in the cancer patient to assess how the disease progression is happening once they have been started with the chemotherapy. So the frailty score, which is having more than 4, so they are having more occurrence of ADRs when compared to the patients who are having less scores. And same like the ECOG performance status also, the patients who are having more than 3 scores, they are having more chances of getting an ADR when compared to less scores. And the CCI, that is a Charlson comorbidity index, the patients who are having 4 to 7, that means high score, they are having more chance of getting an ADR when compared to less scores. So these scores we can assess when the patient has been admitted to the chemotherapy, for the chemotherapy, we can assess clearly and who are having more risk with the help of these scores. So we can give some prophylaxis treatment or the management for those patients in order to prevent the ADRs at the minimum. So that is our objective for this study. So here the predisposing factors like age, gender, underweight, obese, metastasis, and frailty greater than 4, and performance status 2, and comorbidity between 4 to 7 has a more chances of occurring ADRs in cancer chemotherapeutic patients, we can say. And finally, I can conclude that the prior understanding of these factors, so which is causing more ADR occurrence. So actually saying this we have been seen in the lesser patient. That means 2 students, patients only we have been taken, and specifically we have been went for who has been confirmed ADRs. So we have been taken the assistance from the pharmacovigilance associate as well as clinical pharmacists. The patients who are having a confirmed ADRs, for them only we have been taken considerations. But we can say that there is the patient who just come OP, they will take the OP and they will go for the afterwards, and they may get some sort of ADR incidences, but they won't communicate to the prescriber physician. So those are the some of the labs, or I can say some gaps for the study. So if you could able to understand the fact patient, we can make them to understand, we can make them to aware, and we can able to improve the quality of life of the patient, which is the utmost goal of healthcare sector. So I am giving my sincere thanks to Dr. Niranjan Babu sir, Principal sir, and Sunil for the pharmacy, and Kiran Kumar, who is an associate professor in the Department of Medical Oncology. And my happiest congratulations to the Adichunchanagiri sir. Thanks.
What is the output of this? And you are saying that many of the factors that those who are not reported, patients who are under the chemotherapy, who are not reported.
Who are not reported. That means who are not having confirmed areas.
Yeah.
Yes, sir. Actually, it has been started, sir. Now we have been associated with the streams. So we are trying to get more data from those OP patients also.
As per the presentation, I am not the impression that you are still the work is not complete.
Yes, sir. Yes, sir. It's ongoing study, sir. Yes, sir.
And in one part you said about the CCI index, if it is between 4 to 7.
Yes, sir.
Okay, your data is also showing that most reaction is of 7.
Yes, sir.
Okay. I mean, this part you are not carried out with this chemotherapy patient.
Sir,
this study you are not carried out.
They have been carried out, sir. We are doing it, And here we can see means the patients we can see in the oncology ward, the patients who have been means they are some patients who are having severe diseases will give more doses of chemotherapy drugs.
The question was like I was supposed to ask you that what kind of drugs they use and all those things, but that time is not permitted.
Yes, sir. Yes, sir.
Drugs and the type of drugs.
Yes, sir.
All these things, condition of the
Yes, sir. Those things and all I am not saying here. I am just saying my maximum.
Type of cancer means you are taking all the type of cancer patients?
Yes, sir. All the type of cancer patients who are undergoing the chemotherapy.
What type of ADS you observe?
Mostly we have been seen, one thing will be common, sir, for those they are giving some apripitant and all. And apart from that, we have been seen after starting chemotherapy, anemia, leukopenia, and some patients is having a GA bleeding. Anemia and leukopenia mostly caused by cisplatin and doxorubicin and all. And we have been some sort of hematological areas and renal failures. Some cases also we have been seen, sir. Thank you so much. Thank you. Yes, actually, I have shown. Yeah, in our study, the population males are having more impact. But there is one also previous study which is stating that it's some who are having HRT, hormone replacement therapy, the women who are undergoing hormone replacement therapy, and the women who are taking birth control pills and oral contraceptives, right? So they will be having more chances of getting ADRs. So those patients we didn't get. Actually, it's an ongoing study. Thank you so much. Thank you, Honorable.
Today we are going to represent hypothalamus pituitary adrenal axis. I am the final year B.Pharm student studying in Adichunchanagiri College of Pharmacy. I asked this concept because today everyone are suffering from the stress. I am curious about this concept and I decided to take this and I want to know about this concept. So let me introduce the HPA axis. HPA, hypothalamus pituitary adrenal axis. HPA axis is the coupling of hormonal coupling of hypothalamus, anterior pituitary, and adrenal cortex. HPA axis is the vital component for regulation of stress response. HPA axis dysfunction may cause stress, and this dysfunction may lead to the various dysfunctions. Hypothalamus pituitary axis control reactions like stress and regulate the digestion in the system and mood, emotion, and sexuality, etc. Hypothalamus mainly involves in the secretion of corticotropic hormone. Corticotropic hormone released by the corticotropic releasing cells, that is corticotropic cells. It is the main factor to secrete the ACTH hormone. ACTH adrenocorticotropic hormone. In 1981, Hale and his colleagues announced the sequence of 41 amino acid peptide from ovic hypothalamic designated corticotropic releasing hormone, which show greater ACTH releasing potency in vitro and in vivo than any other previously identified endogenous or synthetic peptide. The CRS distribution in the brain and brainstem spinal cord can cause the deleterious effect on the stress. Anterior pituitary gland mainly produce secrete the adrenocorticotropic hormone. These adrenocorticotropic hormone regulated by the corticosteroids. The target organ called the ACTH, which acts as the major regulator of cortisol. Cortisol is the main concept of regulating the stress. Cortisol can be produced by the glucocorticoids, which comes under the adrenal cortex. Adrenal cortex produces the glucocorticoids synthesized in the zona cristallata as the final effector of HPA axis.
Now go for the discussion. Result and discussion.
Metabolism of HPA axis. HPA axis mainly stress, drug, and disease cause a positive feedback mechanism. Here, HPA axis contain 3 types of cells. One is hypothalamus, one is adrenal cortex, and one is pituitary gland. These 3 types of cells produce 3 different hormones. These 3, the stress dysfunction can be modulated by the hormone called cortisol. Cortisol produced by the glucocorticoids hormone. Pathophysiology of HPA axis.
Now go for result and discussion.
Just about the introduction of HPA axis. Finally, I am concluding that HPA axis is the highly complex process that is influenced by both positive and negative feedback mechanism. Many mediators include hormone, neurotransmitters, cytokines, and growth factors have an impact on the functions of HPA axis. Glucocorticoids are the crucial for maintaining homeostasis and allowing the body to anticipate, react to, and recover from physical and mental stress. The imbalance between the glucocorticoid and mineralocorticoid may be contributing factors in the dysregulation of the HPA axis. Thank you.
Shall I start? Good afternoon, everyone. Myself, I am a research scholar in College of Pharmaceutical Sciences, Southern University. Here I am going to present about qualitative and quantitative estimation of nitrosamines in citizen hydrochloride tablets by using gas chromatography mass spectrometry methods. So, introduction. Actually, nitrosamines are the carcinogenic compounds due to the more citizen is a very common OTC drug, over-counter drug. So what will happen? The people you used to take the citizen, the nitrosamine content which is present in the citizen should be less. Otherwise, people will get the carcinogen because of the continuous usage. So to minimize that, the nitrosamines in the drug product should be analyzed and it should be evaluated properly. That every maximum daily intake should be minimized. Then only people can will not get the cancers. So controlling of the nitrosamine in the drug and drug product is very much important. So citrazine synthesis, if we see the nitrosamine formation is very common with wherever the nitrosation is happens. So in that citrazine 9-nitroso-dimethylamine and N-nitroso-diethylamine and N-nitroso-dibutylamine are the possible nitrosamines which can be generated during the synthesis of citrazine dihydrochloride. Hence, an accurate and precise analytical method is required for estimation of these nitrosamines in the citrazine dihydrochloride tablets. Then it will be easily we can control during the synthesis. So this is our aim is to develop and validate a method for the analysis of NDMA and NDEA and NDPA for the cetirizine dihydrochloride tablets. The method validation for the nitrosamines as follows. That is system stability, specificity in the using the tablet matrix and linearity, limit of quantitation and limit of detection and accuracy and precision at LOQ level was done using the tablet matrix. So this is the structure for the N-nitrosol dimethylamine and N-nitrosol diethylamine and N-nitrosol dibutylene. So the material methods used is actually we have purchased the standards NDMA and NDA and NDPA from the Wipro labs, and the instrument used here is GCMS, and the balance we used, and the GCMS is Agilent 7890P model with MS with the triple quad is used with the ALS auto sampler. So mainly drug the solvent used is acetonitrile and methanol. So here materials and method. The GCMS method, this involves the GC column with the BX backs, MS column was used, and collision gas was used for nitrogen, and quenching gas is helium, and injection volume 2 microliter with the split, and this is the ion source for 230 degrees Celsius, and the mainly it is involved with the mass spectrophotometry analysis. So single ion monitoring mode was used, that is SIM mode. So NDMA that mass is 74 and the NDEA is 102 and NDPA is 158. And the solution standard preparation, the mixture of the nitrosamines was prepared, and that 200 ppb stock concentration was used. Then further it was diluted to 30 nanogram per ml using that was used as a system stability standard, and further that was diluted to LOQ level is 10 nanogram and LOD level is 5 nanogram per ml. And the sample preparation was the required amount of cetirizine dihydrochloride tablet was weighed, and it was acetonitrile was added and water for 2 minutes, then centrifuged at 4500 rpm, and the supernatant was used for the GCMS analysis. So the experimental parameters, whatever the results are here, the system suitability was meeting the acceptance criteria for the RSD of the area is lesser than the 10%, and the specificity, there is no interference of the cetirizine IAPA, and as well as the any other excipients is not interfered with the nitrosamine peaks, and the linearity was 2 ppb to 100 ppb, and the limit of quantitation is it is 10 nanogram per ml, that the signal to noise ratio is more than 10, and limit of detection is that level is 5 nanogram per ml, that is also the signal to noise ratio is more than 10. Then the accuracy and precision at LOQ level which was performed having the good recovery 99% to 70%, and the RSD limit also it is not more than 30%. So these are the actually chromatograms. The standard chromatograms contains the all NDMA, NDPA and NDEA, and the LOQ chromatogram also having the signal to noise ratios, and LOD chromatogram. Blank does not have any chromatograms over there. Then the cetirizine dihydrochloride tablets also it is analyzed. In that also we have not found the peak for the these nitrosamines. So the cetirizine tablet which we used for the analysis does not have the nitrosamines. Discussion is nitrosamines are more correctly speaking, N-nitrosamines are referred to any molecule containing the nitroso functional groups. These molecules are the main concern for the probable cause of the human carcinogens. So determine the N-nitroso diethylamine and N-nitroso dimethylamine and N-nitroso dibutylamine in cetirizine tablets. GCMS method was developed, and the method was verified using the different analytical experiments parameters. Then the analysis of the cetirizine dihydrochloride was carried out, and it was giving the good results.
Okay, madam.
Yeah.
Cetirizine was of a racemic mixture you have taken, or any isomer of the cetirizine you are using?
No, sir, there are no isomers of cetirizine. Actually, cetirizine dihydrochloride acid tablet only taken. So, in that only the nitrosamines are spiked, and it was extracted and injected through GCMS analysis.
GCMS spectrum are not seen. You have taken the GCMS spectrum.
Actually, that is the chromatograms are from the GCMS only, sir. Actually, when we are doing the scanning, after the scanning, we will get the mass from the GCMS. Then that mass we will put it in the selective ion monitoring mode. Then we will get the chromatogram as such. So this conclusion is this method is whatever developed is very much useful for the analysis of cetirizine. Any nitrosamines is available in the cetirizine or not, it can be validated method also. So anybody can use it.
What is the mobile phase used here?
Here mobile phase is that carrier gas only, sir. Nitrogen and helium. Quenching gas is helium and carrier gas is nitrogen.
So they somewhere about acetonitrile and methanol.
Acetonitrile and methanol is for the diluent purpose.
National anthem.
Yeah, thank you all. I hereby want to acknowledge my college, my guide KB Prema Kumari, and this podcast I have carried out in vitro research solutions. I want to thank them also. Thank you all.
Good evening, all. My name is research scholar in ICIAPS. My today presentation title with neuropharmaceutical investigation of Dudinia viscosaline based on experimentally induced So we said, what is the efficacy of the plant extract Dudinia viscosa on Adichia mice, as described by Dr. Bandrich, the OJ of Alzheimer, where the accumulation of data and the genes like receptors and listening and the use of those plant helps in neurogenesis, also because they have the strong antioxidant, anti-inflammatory, and neuroprotective. We can use them for treatment of the Alzheimer's disease. So for the aim and objective is the objective of the studies, we have to use the ethanoic extract of Dudinia in behavior models. We use elevated plasmase and modus fraternase, and we have estimated the whole brain acetylcholinesterase. Here is the material. We have collected the plant, indicated, and it was being extracted using ethanol. No.
Okay, just put it this results of discussion. Summary, yeah, summary and
See, in summary, we have used here
evaluated by using the extraceptive and interoceptive, the elevated plus maze and the Morris water maze. And we have to calculate the escaping latency and the transfer latency of the plant. So the plant is showing very nice learning and memory. And once the models has been finished, we use the acetylcholinesterase in the brain. Acetylcholinesterase is an enzyme that causes Alzheimer. So it was very strong inhibitor for the acetylcholinesterase. This is the conclusion.
So you have carried out the experimental part of it?
Yes, it was done. And further studies in.
My question is that why did you select directly the leaf from the particular plant? Why not for the stem or bark? Or what was made you to go directly to the leaf part?
It's related to the review literatures because they have the highest polyphenolic compounds, the stronger antioxidant and anti-inflammatory. So many literatures we use in the leaves. Mainly it has the anti-diabetic drugs, and the further studies we want to use what is the efficacy of diabetic and cognition. Diabetes usually affects cognition.
Okay.
So further studies we are going to do in that, and what is the efficacy on diabetic mice for cognition. This is. Sorry for that. It was too
Thank you.
Thank you.
Thank you very much. Padma.
Your email, the one I sent.
Volunteers, delegates, ask them to sit inside. In 10 minutes, we will wind up.
I request all the delegates to enter the auditorium. We start the valedictory function shortly. If any of you are interested to give any feedback, you can come forward and do. Any of you.
Okay, good evening, one and all. Myself, Snehal Sakorkar. I am working as an assistant professor in the Diva Patil College of Pharmacy, Pune. From the last 2 days, we are here and listening so much energetic speakers. And what I found actually, it was quite difficult after the COVID to come forward and organize such a big event. And it was quite challenging to everyone because organization of the guest and collection of that subjects and everything. But what I actually exactly found that very best time management is there. Means very fine at exactly time the event is started and the way they are coordinating the event and the volunteer students, they are working very well. And from the registration onwards, if I found any problem related to because Pune and this region is very long distance, so I'm 100 times I'm calling that ma'am. That ma'am, I'm facing this issue. Ma'am, where exactly it is located? Then it suggests me near hotel. So without any hesitation, ma'am is always there to guide me. So I found very best. That's why I got some of the more confidence then rather than attending the virtual, why should not I attend exactly the place itself? So it was very good experience to come here and interact with the people. Still there is a language barrier, but I didn't feel it is because of all of you are very here to support everyone with delegate who have come here. Thank you everyone. So it was a very wonderful organization. I am thankful to organization team itself. Thank you.
Thank you, ma'am.
Great to respected as. So I am Rasha Pradesh Shawans, working as an assistant professor in Kelly College of Pharmacy, Belagavi. So last 2 days, I am presenting here only. I am seeing a lot of the poster presentations and the research work presentations. It was very nice and good. And the speakers also is in a very high profile people. And I want to suggest to the students, take the opportunity to the respective speakers, what they are given the presentations of any keynote speakers, and go to the proper way. In the small thing, the innovations it was having a lot of research work is there. Kindly focus it and concentrate it and move on toward the research innovations in the pharmaceutical science. And thank you very much for giving us an opportunity to give us an to the poster presentations. And thank you all the volunteers and the respective faculties, what they are working the very tough to conduct the conference. I know about that, all those things. So all the best. Thank you.
Thank you, sir.
Everyone, myself Pooja. I'm currently pursuing PhD in Sri Adichunchanagiri College of Pharmacy. So first of all, I'm very happy to take a part in APR. And foremostly, I want to thank our Dean of Pharmacy, Dr. Ramesh sir. So, and the communication and the bonding between APR and our institute should go on for a long time. So, and the one request is that if you conduct this, please try to increase the time. 5 minutes and 3 minutes is insufficient to give a presentation. That is the thing I want to say. And once again, thank you. And the people who came from a long distance, once again. And the foremost thing is that all the best for the future and do a very good research. Thank you. Thank you.
Thank you, ma'am.
Jai Shri Gurudev. Never be afraid to go after your dreams.
Jai Shri Gurudev. Never be afraid to go after your dreams, regardless of the negative forecast that has been predicted upon us. Beat the odds and let us shine. Life is not fair. We must keep striving for success.
We have all gathered here to mark the end of the conference, the valedictory. I welcome Dr. Umesh Manakar, Mr. Raja Agnihotri, Mr. Rudrabanu Satpathy, Dr. B. Ramesh, and Dr. Prakash Godandavar onto the dais.
Now, I would like to welcome Dr. B. Ramesh, Organizing Chairperson, Dean and Principal, Sri Adichunchanagiri College of Pharmacy, to give the highlights of the conference.
Jai Shri Gurudev, my humble pranams to the lotus feet of Honorable Chancellor, Sri Dr. Nirmalanandanath Mahasobaji. I know you are all eager to go back to your hometown. Nevertheless, this is a time for the consolidation and brief you about the 2 days deliberations which we had and a wonderful 2 days conference, international conference. First of all, my sincere thanks to all the delegates, around 427 delegates who are really looking for this type of scientific deliberations. A big round of applause for all the registered delegates. And as you know, 262 delegates were present here physically, and virtually 165 were online. This shows the commitment of all those delegates who really try to contribute for the profession of pharmacy. A big round of applause for all of them. And as the faculty of D.Y. Patil, College of Pharmacy Pune was mentioning, there were so many challenges in organizing the real-time conference after this COVID and all. But nevertheless, the team at Sri Adichunchanagiri College of Pharmacy have done a wonderful job, local organizing committee. My special credit and thanks to all of you, all the staff members, and big round of applause for all the faculty. And it goes without saying the ruthless efforts done by Dr. Prakash Gowdanavar for the last 4 years, last 4 months. He was continuously collaborating with the APR people. A big round of applause for him. And, we have with us eminent CEO of the APR, Mr. Rudrubhanu Satpathy. My special thanks to you and your team members. Please, we'll try to continue the same trend of innovation, collaboration thing in days to come, so that our student fraternity will get the more benefit. And you have seen in the last 2 days, you have seen 8 keynote speakers, starting from Roshan, Madhav Rao, Dr. Banakar, and Dr. Maniam, Dr. Rajesh, Raja Agnihotri, Devendra, and Dr. Karthik. So you have gotten an insight and exposure how the pharmaceutical industry works. Special thanks to the one more speaker, Raja Agnihotri. In 4 or 5 slides, he has put all the pharmaceutical industries and business in a nutshell, and he has explained. It was really an eye-opener for the administrator like us. Really, I thank him also, Raja Agnihotri sir, for your wonderful presentation. And I thank all our teaching and non-teaching faculty members for their contribution for making this 2 days national conference a successful one. And I also thank the media persons and the technical crew who made this possible. Thank you, one and all.
Thank you, sir.
Here we come to the most awaiting moment. I would like to call upon Dr. Prakash Gowdanavar to announce the prize winners.
Jai Shri Gurudev, respected dignitaries on the dais, faculties, and dear delegates. Very good evening to everyone. Before announcing the awards, because still the physical awards are getting ready, as oral presentations were delayed by some time. What I'll do is, they're coming, sorry. Okay. I'll just announce the virtual awards, the delegates who presented on virtual mode. So the awards I'll announce for the virtual e-posters as well as e-oral. The first prize is given to 2 delegates. That is Dr. Girish Pai, Manipal College of Pharmaceutical Sciences, Manipal. Again, first prize, Karima Ambreen from Kuvempu University. Second prize, again in virtual e-poster and oral presentations, Archana Devi Ashok Kumar Patel, KM Kundanani College of Pharmacy. Again, second prize, one more delegate, Rahul Sachdevo, Krishna Vishnu Vidyapeeth College of Pharmacy. Third prize, E-Oral, 2 members again, Karthik R from JSS College of Pharmacy. Third prize, one more delegate, that is Sharija Hijaji. He is from University of Kashmir. Now I'll announce the virtual e-poster awardees. The first prize again for 2 members. Mansi from DY Patil Institute of Pharmaceutical Sciences. One more prize for the, in the first place, Shanaj Banu from Dayanand Sagar College of Pharmacy. Second prize in e-poster, Sonal Dubey from Dayanand Sagar College of Pharmacy. Again, one more member for the second prize, e-poster, Sandra KS, Enepoya Pharmacy College and Research Center, Mangalore. Third prize in virtual e-poster category, Kanj Mudalwan from SRM, Chennai. Third prize for one more member in virtual e-poster category, Lata Kotapalli, DY Patil Institute of Pharmaceutical Sciences, Pune. For all these virtual presenters, e-poster will be made to them. Okay, thank you. I'll come on to the physical mode awardees. Yeah, we'll start with the oral presentations. Again, there are 2 prizes in first position, 2 prizes in second position, and 2 prizes in third position. First prize in oral category goes to Pandrish MD from Adichunchanagiri University. I request dignitaries on the dais to present the certificate and the medal to Dr. Pandrish. Big round of applause, please. Congratulations, sir. Again, first prize in oral, Snehal Shashikant from DY Patil College of Pharmaceutical Sciences and Research. Again, this request all the dignitaries and the dais to present certificate and medal, please. Congratulations, madam. Now we will continue with the first prize in poster presentation. There are 3 prizes to be given in the poster category. The first prize, first candidate is M. Mamata from Seven Hills College of Pharmacy. Congratulations. I'll read out, please come onto the dais. B. Prakash Kumar from Sri Adichunchanagiri College of Pharmacy. One more first prize, Sriram D. Ranade from KLE College of Pharmacy, Telangana. Take a round of applause, please. Who got the first prize, please join for the photo. Oral, Pandrish and Snehal, please join. Moving on to the second prize, both in oral as well as post category. Read out the names of the members for oral awards in the second place. KM Jayasri, Seven Hills College of Pharmacy. Munajila from Sri Adichunchanagiri College of Pharmacy. Poster presentation, second place, again goes to 3 members. Vismaya Achyuta from MS Ramaya College of Pharmacy. If anyone from MS Ramaya, please collect the prize. Again in poster, second prize, Purushottam KN, Sri Adichunchanagiri College of Pharmacy. One more prize for second place, Young Shini Cherishna from Nirmala College of Pharmacy. Big round of applause. Moving on to the next prize awardees, that is third prize in oral as well as poster presentation. The first 2 prizes for oral in third place. Binu Chetri from Alamin College of Pharmacy, Bangalore. And Kaushal V from MS Ramaya College of Pharmacy, Bangalore. In poster category, 3 awards again in third place. Gajanan from HSK College of Pharmacy, Bangalore. Akshita from Sri Siddhanagar College of Pharmacy. Asha BR from Sri Adichunchanagiri College of Pharmacy, Vijayanagar. Anyone from Sri Adichunchanagiri College of Pharmacy, please come and collect the prize. Big round of applause for all the prize winners. Yeah, that completes the award giving moment. Now, it's my honor to greet my people. Of course, organizing secretary, as organizing secretary or organizing chairperson, Dr. B. Ramesh sir. We need again help from the different committees. So now it's a moment to honor all the committee members. To start with the organizing committee, Dr. Yunus Pasha. Sagir Ahmed. And I'll read out the scientific committee members. Of course, the presentations, both oral and postal, were marvelous. The nice arrangements in all the rooms. So my heartiest appreciations and congratulations to all the scientific committee members. Dr. Anegowda HV, Dr. Shankar Mani, Navin Nagre, Next, hospitality committee, of course, we also strive for more than a month to arrange the accommodation for all the delegates coming from the different parts of the country. Dr. Rupesh Kumar, Mr. Amrutanand Tripathi, Mr. Purushottam. I will thank once again to all these committee members who have relentlessly strived to have this international conference at Vijayanagar campus. Thank you all. Now it's time to appreciate our moderators, two beautiful girls and one gentleman. Since again 1 week we were involved in writing the script. I always tell them to correct this, correct that, but still the script was ready on the time. So I request again our the guest on the dais to appreciate Miss Lohita. Mr. Lakshmi, technicians. I think she's not there. I request Ms. Thank you. Thank you very much.
Congratulations, winners, and a big congratulations to all our participants.
Now, I would like to call upon Mr. Raja Agnihotri for the final remarks.
After Ramesh speaks, we need to adjust a lot of things. Beginning up, it was very learning from the beginning, and the big list goes of thanking. First is that we have been part of many international conferences, and where I have been as an organizing committee, sometimes scientific committee, sometimes coordination committee, many things. And I know the pain it involves, and coordinating with the delegates and the speakers. And always the speakers will not have time. They say only to yes by giving a slot, but after that they don't. It's a challenge, even including me. So you did all a wonderful job, and very importantly, one by one I'll go. First thing is that the concept of this international conference is been there with Dr. Ramesh for a long, long time. Because I'm a frequent visitor here, so he keeps discussing. And the way it has been done, just to put on a brochure. The first thing I said that don't put on the brochure just as an international conference. It should be an international conference. And he ensured that it is on par like an international conference. And he ensured that the conference happened. So first thing is that, thank you. And I know that in an educational institution, it's you have to move the rocks. It's a rock. You have to move the rock. Sometimes you have teams, sometimes you have to push the rock on your own. And every time you move, it will come on you. Right? That's what it happens. And when you are organizing such a big event like that, but you don't have to leave. Slowly, you have to build a team to pull the rock. So he built a team. Right? And I thank you for that. And another thing is that the first time I asked him is that, why do you want to do an international conference? We in the industry do it because we get mileage, we get reach, and our business will improve. Definitely. And I want to reach more clients. So that's how it is. He told me one thing, my students should get exposure. They should get. See, that one spark was enough for me that really there is a fire in the belly, and he did it. And thanks to all the students, you may not get this opportunity again and again to get such a kind of exposure what you had in these 2 days, and including the virtual, such a beautiful organized. So APR, BioLinks, and United Innovators who are there, wonderful job. And in fact, in the lunchtime also I was telling, where I was many international events, I was a speaker. And when we go virtual, first 15 minutes goes, am I audible? Can I unmute? Please ask somebody to mute. Cooker is whistling in the backside. So these are the common things. But here, the voice was audible, picture was clarity, even the questions were asked. So excellent. It really looked international. Thank you. Thank you for that. And the staff involved, that is organizing committees, all transport committee, coordination committee, and all those things they have done wonderful job. And I could hear from one of them, from Deva Patel, saying that accessibility to the people, to the staff, whenever they want, they picked up phone, they guided us. And I was told that some were put up in Yediur, and from there the transport was arranged to come. Excellent. That's how the students should be taken care, and your vision will be met. I like that vision, and he's sticking to that. produce globally competent pharmacists with skill, knowledge, and attitude. I think they are in the right direction. So please keep up that work. Excellent. And third thing is, I thank all the keynote speakers. I know most of them, and it was my dream come true to meet Banakar sir. After a very long years, I met him. And if this podium or this platform would have not been there, we would have met in a different, but I met him here. Thank you for meeting us and for the students. Why I thank the keynote speakers is the intention to tell to the fraternity of the students and the research team, because they are going to be the future of giving the global quality products. Until we don't share our experiences, good or bad, it will not help them. So I thank all the keynote speakers to come here and deliver or talk or share whatever they know. And for all the speakers, one person was consistently coordinating, that is Dr. Prakash. And always, he used to ask me, can I call you? Do you have time? And whatever time I give, he used to respond. And so also Dr. Ramesh. So I thank them for the patience they have with dealing like characters like us. So that was with an intention to do a good job. So that was wonderful. And some presentations I could see, some mind-boggling presentations I could see, some mind-boggling sparks of research work I could see, especially the protein crystallization with some discussions I saw it. And very beautiful ideas you people have. Please pursue it. I thank all the as well as virtual as well as physical poster presentations. Wonderful concepts and thought process what you people have, whether it is good or bad or innovative, you present it. You don't know, somebody will pick up that idea and do something wonders with that. And thanks for all participation. At our time, we used to have hesitation to talk also. You people are coming all the way from different corners. Even Kashmir also, I saw somebody getting an award. They coming and presenting it. It's great. And all posters were good. All present. I have made some notes because it gives me spark to think and tell to the industry people that there are some things happening here. And I thank all of them. And especially the AV systems, I thank wonderful sound systems as well as presentations. There was not much of hassles. And very importantly, the non-teaching staff, I should tell them because of the food arrangements, time to time tea for me, right? I keep on asking, time to time water for me, I keep on asking. So they gave that, and not only tea, it was very tasty. I think you all enjoyed that. And very beautifully, see, this is what I like with Dr. Ramesh. He goes very minutely. When he's serving food, he ensured that there is a shade in the top. You sit, aram say, and eat. So that shade also he took care. Usually it will be open area in the sun, we eat with sweats and all. He ensured that they sit and eat and tables were arranged and all. So very minutely he has gone into the things, and I really appreciate and thank. And lastly, I thank all the parents for allowing you to come here. Right? Unknowingly, they have sent you, and that could be because you would have told you are going to Sri Adichunchanagiri College, which is a big mutt where Swamiji stays. Please also go there, put your footsteps there. You will also get the blessings of that holiness. So that could be one of the things. So you have all visited a very holy place, gathered knowledge, presented, shared. I think God will always bless you and you will always do well. Right? Thank you all for participating. And I think I will bounce with many of you in the industry. Right? And do well. Keep researching, keep innovating, keep thinking. You are not in the wrong industry, you are in the right industry. God bless you all. Thank you for the time.
Thank you, sir.
I request all our college teaching staff to come over the dais after national anthem for a group photo. Just a thank you is a mighty powerful prayer, says it all.
Now, I would like to call upon Dr. Rupesh Kumar to propose the vote of thanks.
Jai Shri Gurudev. Very good evening to all ladies and gentlemen, dignitaries on the dais, and with the divine blessings of Puja Swamiji, on behalf of the family of ICIAPS, I want to thank all the organizing committees. And before that, to begin with this, I want to thank our management as well as Swamiji and our beloved principal, Dean, for constantly supporting and encouraging us to conduct such a great event, international conference for the first time in ICIAPS under ACO umbrella. So with this special notes, I want to thank the organizing secretary, Dr. Prakash Lodana, and constantly supporting all the team and leading us every day, every minute, every second to take it as a grand success. With this, I can thank even organizing committee as well as Dr. T. Y. Pasha sir, Bharti madam, and scientific committee, hospitality committee, including myself and my team, and audio visual team. And apart from this, the most important delegates from different parts of our country. So I want to thank on behalf of our family for attending this two international conference. Once again, I thank each and everyone. Thank you, sir. Thank you.
Just one note for every delegate. I think the feedback link is already shared with you. Please give us the feedback for any improvements. Please. I think everybody has received the feedback link. Please submit the feedback link. Thank you. I think it is already, yeah, it's projected. Please do this for us. Thank you.
Thank you, sir.
So I guess it ends here. We'll go our separate ways and hope that we'll see each other somewhere in the future. Thank you one and all. May I now request all to raise for the national anthem.
Hello. I request all the delegates to come to a group photo.
Are you ready?
Due to the want of time, volunteer certificates were not given. We'll hand over it on Monday or Tuesday. Thank you.
Hey, Pawan, असली चेंजर आहे त्या पडी मिल ठीक आहे